Knowledge is the light that guides us through the darkest paths, the wealth that grows when shared, and the key that unlocks endless possibilities. Good morning and a warm welcome to all. It gives me immense pleasure to be here today as we inaugurate IPTCON 2024, a two-day international conference on recent trends in drug development and translational research, organized by the esteemed Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalayam, Tirupati. This conference stands as a testament to our collective pursuit of knowledge and innovation in the pharmaceutical sciences. I, Dr. Sheikh Asha Begum, alumnus PhD student on behalf of the entire team of IPT, take the immense pleasure to extend a heartfelt welcome to all the dignitaries, esteemed speakers, distinguished guests, scholars, and students who have gathered here in this vibrant seminar hall. Let us embark on this journey of intellectual exploration, exchange of ideas, and collaborative discussions with optimism and enthusiasm. Ladies and gentlemen, this morning we have the privilege of being in the presence of extraordinary minds and visionaries who have been instrumental in shaping the field of pharmaceutical research and education. It is my honor to invite them onto the dais. Let us extend our warmest applause as I call upon these esteemed dignitaries.
महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दितं महागणपतिम् महादेवसुतं महादेवसुतं गुरुगुहनुतं महादेवसुतं गुरुगुहनुतं महरकोटि प्रकाशं शान्तं महाकाव्यनाटकादिप्रियम् महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकप्रियं महाकाव्यनाटकादिप्रियं मूषिकवाहनमोदकप्रियं महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि वसिष्ठवामदेवादिवन्दित महागणपतिम्.
Dignitaries on the dais and off the dais, and my dear participants, a very good morning to all of you. Let divine's grace be showered on all of us on this event. That is the supreme energy that guides us always. So it's my honor and privilege to welcome you all to IPTCON 2024, hosted by Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalayam. And we are delighted to bring together such an illustrious gathering of professionals, researchers, and practitioners in pharmacy and Department of Life Sciences. First, I would like to
It's now my pleasure to invite our special invitee, Dr. T.V. Narayana sir, a revered figure whose unparalleled contributions to pharmacy and healthcare have left an indelible mark on the profession. Sir, your presence enriches this conference and we are deeply grateful. Please do honor us of taking your place on the dais.
First, I would like to extend a heartfelt welcome to our distinguished special invitee, Dr. T.V. Narayan Garu, whose inspiring presence truly graced this occasion. We are deeply honored to have our chief guest, Professor V. Uma Garu, our Honorable Vice Chancellor in charge, and President of this function, Professor N. Rajani Garu and Professor P. Sujatamma Garu, Dean of School of Sciences, whose unwavering support has made this event possible. A special thanks to our keynote speaker, Dr. D.R. Krishna Garu, for joining us to share their invaluable insights and expertise. We are also grateful for the guidance of our honorary advisor, Professor R. Nagaraja Garu, and the dedicated efforts of our organizing secretary, Professor M. Vidyavati, whose hard work has brought this event to life. Lastly, I warmly welcome all participants, scholars, and professionals joining us from across the globe. Your presence reflects the collective dedication to advancing pharmacy practices and fostering meaningful collaborations. It is both an honor and a privilege to stand before you today and introduce one of the India's most prestigious institutions, Sri Padmavati Mahila Vidyalaya and its flagship Institute of Pharmaceutical Technology. Sri Padmavati Mahila Vidyalaya was established in 1983 in Tirupati, that is in Andhra Pradesh, by Sri N.T. Ramarao Garu. The then Chief Minister of Andhra Pradesh, was founded with the goal of empowering women students to become effective contributors to the nation and to develop their leadership skills in various fields. SPMVV holds the distinction of being the second women's university in India, and it is the only women university in Andhra Pradesh, underscoring its unique role in promoting women's higher education. This university is accredited by NAC and has achieved an A+ grade, reflecting its high standards in education and overall excellence. SPMVV is ranked among the best in various national and international rankings, including NIRF, Times Higher Education, QS, ISO certifications, and the Green Metric ranking, further establishing its commitment to sustainable practices and academic leadership. SPMVV is the only university in Andhra Pradesh to receive 100 crore grant under the PM OSHA initiative. This grant will be used to strengthen infrastructure, promote research, and elevate the university as a leader in women's education in India. IPT has forged strong partnerships with leading national and international institutions and industries. This includes collaborations with universities and research organizations in countries such as the UK, USA, Malaysia, Bangladesh, South Korea, and Japan. Additionally, IPT sends students for science exchange program like Sakura in Japan, providing them with invaluable international exposure. Recently, our students have selected for global undergraduate exchange program sponsored by US Department of State of Bureau of Education and Cultural Affairs. So about the infrastructure and research facilities, IPT is equipped with modern laboratories for pharmaceutical chemistry, pharmaceutics, pharmacology, and microbiology, supporting both academic learning and research. The university also operates 3 incubation centers and 7 research centers, including rural technology parks, designed to promote innovative projects and rural development solutions. So about the library and e-sources, the university's library offers a vast collection of textbooks, research papers, journals, and digital resources to support students and faculty in their academic and research pursuits. So about the Institute of Pharmaceutical Technology, the Institute of Pharmaceutical Technology was established in 1995, is a key academic unit of SPMVV, offering comprehensive programs in pharmaceutical sciences. These include B-Pharmacy, M-Pharmacy with 5 specializations in pharmaceutical chemistry, pharmacology, pharmaceutics, pharmaceutical analysis, and pharmacognosy, as well as PhD programs, both for national and international students. Our senior most professor, KVSRG Prasad Garu, your presence is very valuable, sir. We're very lucky to have you in this conference. So he works hard for the development of this institute. Very hard, very hardly he works. About the PhD achievements, the institute has awarded 1 or 2 PhD degrees, reflecting its commitment to research and innovation in pharmaceutical sciences. IPT has earned a place in the 60th rank nationally in MHRD NIRF rankings for pharmacy, further highlighting the academic excellence and research output of the institute. This institute is approved by PCI and boasts a dedicated and highly qualified faculty. About our research funding and collaborations, IPT has successfully secured research grants from major national and international organizations, including AICTE, UGC, SAP, DST FIST, DST SUB, ICMR, and DBT Builder. These grants support groundbreaking research in pharmaceutical sciences, from drug development to biopharmaceuticals. So about our publications, patents, and innovation, IPT has been at the forefront of innovation in the pharmaceutical field, publishing above 50 patents in areas such as drug delivery systems, therapeutic agents, and pharmaceutical formulations, demonstrating its commitment to pioneering advancements. IPT published above 600 papers and around 500 presentations with high H index value. Many of our teachers received state Best Teacher Awards, Best NSS Program Officers, and Best Research Awards. So about our students' achievements, students from IPT have consistently excelled at regional, state, and state and national level youth festivals, sports tournaments, and NSS camps. They have also garnered prizes in various prestigious platforms, highlighting their all-round capabilities. Pharmacy students from IPT have been recognized at the AP Pharmacy Council Awards and have secured top ranks in NIPER as well as in GPAT, demonstrating their academic prowess. About our future plans and vision, with the support of PM Usha grant, IPT plans to enhance its research infrastructure and introduce cutting-edge research programs in biopharmaceuticals, nanomedicine, and personalized medicine. IPT continues to advance its commitment to green chemistry and sustainable pharmaceutical practices in alignment with SPMVV's environmental goals. This institute aims to deepen its international collaborations, offering more exchange programs and research partnerships with institutions across the globe. The Institute of Pharmaceutical Technology at Sri Padmavati Mahila Viswavidyalaya stands as a leader in the field of pharmaceutical education and research. With its robust academic programs, cutting-edge research initiatives, global partnerships, and entrepreneurial ecosystem, IPT is well positioned to contribute to the advancement of pharmaceutical sciences, while also empowering women in the field of higher education and innovation. The future is promising, and IPT remains committed to fostering a culture of excellence, creativity, and leadership in the pharmaceutical sector. So our theme for this conference is recent trends in drug development and translation research. This highlight innovative approaches aimed at accelerating the discovery, screening, and delivery of new therapies. One significant trend is the rise of precision medicine, which tailors treatments to the genetic profiles and individual characteristics of patients. Advances in genomics and bioinformatics are enabling researchers to better understand the disease mechanisms leading to more targeted therapies. Another trend is the increased use of artificial intelligence and machine learning to optimize drug discovery, predict drug interactions, and improve clinical trial designs. This event will provide an opportunity to all budding researchers, faculty members, industry members to learn the recent developments on innovative drug design and discoveries, cutting-edge biomaterials, and novel drug delivery devices that promise to enhance the therapeutic efficacy and safety profiles. Thank you for your attention. Thank you, Ananda.
Dignitaries on the dais, delegates, invitees, faculty, students. I would like to take this opportunity to very fondly remember Professor Bharati Garu, who has laid the foundation, very strong foundation to the Institute of Pharmaceutical Sciences. I think we need to give her a big clap. And I'm also thankful to Professor Prasad for being here this morning, taking his time out and coming. Now, Professor Rajani said, what did you say? I think she said I have to share my expertise or views on this. I'm sorry, I'm not an expert on the topic because I'm a social scientist by nature, but I'm a little familiar after I have discussed with Professor Vidya and Joshna in terms of what they intend to do in the two-day conference. And also because during the last 2 years, we have been working on transdisciplinary research. And I was coordinating the research on the campus, which was funded by the UGC. And we had a good number of faculty from IPT to work on the transdisciplinary research, which was related to anemia. And we were talking about how the community, the people who are affected by anemia really matter a lot when you want to, work in the area of anemia. So it's not just sitting in the lab and working towards having iron capsules or delivering it to them, but also focusing on why they don't care for acting on anemia, which is a psychological issue, which is also a social issue in the community because it really doesn't matter to them. But in the long run, it does have a lot of impact. So it is from that context that Professor Vidya said, I can be here and I can say a few words. But I said she has to give me a note on what the topic is about. So the basis for my talk is the note that was given by her. I'm happy to be here, particularly because it is the conference of the Institute of Pharmaceutical Technology on the campus, which is one of the good institutes. And they have achieved a lot over the years. In terms of specific ranking in IPT, they've been doing very well. And the contributions coming from the faculty, research scholars, and the M-Pharmacy students have contributed a lot in helping the university to get a good rank in the NIRF. And we have been improving over the years. So today we are fit to receive funds from the ANRF, Anusandhan National Research Foundation, which was established by DST, a new move based on the NEP, New Education Policy, because all research funding is now going to come through that. We are not a hub yet because I think 25 and below rank are only eligible to be a hub institute, but we are working along with BITS Pilani and Hyderabad campus. So there's a long way to go. We are working towards it. And as Professor Joshna just mentioned, we have also been identified to receive the 100 crore grant from the UGC under the PM Usha program. So good amount is dedicated to research and about 20 crores to establish new labs and buy scientific equipment. Now, IPT is also getting a new building, which they deserve under the PM Usha program. And they will also focus a lot more on the research. I think they plan to work on nanotechnology and other aspects. And I'm sure drug delivery is one of the important aspects that they would like to focus on. So having said that about the institute, I once again welcome you all to this conference. I think Dr. Krishna Garu is the key person, the international person who will be delivering the keynote address and the 2 days is going to focus on recent trends in drug development and translational research. So I am given to understand that the critical process in drug development is central to turning scientific discovery into meaningful treatment options for patients. That is the journey from the bench in the lab to the bedside of the patient and trying to establish a link between these two rather than working in isolation in a lab, trying to develop some drugs that are useful for an unknown person. So the process of taking the drug from the laboratory to the clinical application is long, yes. It is also very complex because there are a number of aspects and factors that need to be taken into consideration. And we are not free from challenges. It's not going to be easy at all. And I think it is a continuous process. There's no end to it. Therefore, we need to look into what happens in this particular process. As a qualitative researcher in social sciences, I do very strongly believe that the processes are very important. And these are the ones which will lead to the outcomes of the research. So drug development is heart of improving the global health, and millions of people around the world, they are suffering from diseases that are either poorly understood or lack effective treatment options. So translational research, it is expected, would bridge the gap between the laboratory science and clinical applications, ensuring that new discoveries translate into real world solutions. I'm not sure what these real world solutions are because we keep hearing to so many outcomes and so many impacts and adverse effects also. Particularly, I'm shocked by the vaccination and the type of discussion in the time of Corona. I think Pfizer is the one which has been put to target, if I'm not mistaken. And I really need to know a lot more about it. But what is it that is happening? Though on the other hand, we are talking about real world solutions. Is it the quick time that we need to respond, or is it a real drug, real solution that we need to find? I think there is a challenge between these two. In time of Corona, I think uh, quick response was what was required. But then today we are seeing what is happening as a result of the quick response. Is it a real world solution, or where are we taking all the people who blindly believe these companies who develop the drugs that we take? Particularly when I am one person who will, who don't prefer to see a doctor to take, not to take any medicines. I've taken 1 week to see a doctor to, take an antibiotic before because of the back cold. Now, I say this because uh, people today run to doctors even for minor ailments, which was not the situation earlier. There used to be a lot of home remedies, which we are giving up, which we are not following at all, which have been very effective without any side effects. But today we believe the doctor so much. And on the other hand, we look at the commercialization of the treatment and the prescriptions that are taking place. So this concerns me a lot. And then the other thing that Vidya told me with regard to talking today is the use of artificial intelligence. Again, the abuse. There is a good side of the artificial intelligence. There's no doubt about it. But to what extent do we really need artificial intelligence? She says it is a game changer in the recent years of drug discovery because of the application of artificial intelligence and machine learning. So these technologies allow us to process vast amounts of data at lightning speeds. Yes, again, that is the thing. The lightning speed is, I think, what is more important. And identifying new drugs faster and more accurately. I don't know if they go together, faster and more accurately. I leave it to you all to discuss in the next 2 days with the possibility of this. So artificial intelligence is being used to predict how molecules will interact, optimize drug structures, and even repurpose existing drugs for new disease. So AI-driven drug discovery platforms are already responsible for bringing drugs into clinical trials that would have taken much longer than if it was identified through the traditional methods. So I think it is in this context that the deliberations during the next 2 days are going to take place. I'm really hopeful that the deliberations will bring out new insights, and the young pharmacists to be who are sitting here will gain some knowledge and really engage themselves in research, taking into consideration the needs of the humankind in the true sense, not really focusing only on being very fast, but also taking into consideration the psychosocial needs of the people who are our target audience. So I'm hopeful that you'll explore some of the key trends that shape the future of the drug development in the 2 day conference. And I wish you all the best. And I'm really thankful to both the speakers who have agreed to be here this morning and share their experiences in the area that has been identified for the 2 day conference. I wish you all the best and hope that it is a good success. Thank you.
Respected dignitaries on the dais, off the dais, my dear delegates, and my dear students. A very good morning to all of you. I am greatly honored and privileged to introduce Dr. D.R. Krishna, today's keynote speaker, an eminent scientist, a passionate teacher, and a clinical pharmacologist. Sir received B.Pharm degree in 1976, M.Pharm degree in 1978 from Andhra University, PhD in clinical pharmacology in 1984 from Kakatiya University. Later, Sir completed postdoc from Dr. Margaret Fisher Bost Institute of Clinical Pharmacology, Germany. Sir joined as assistant professor in the year 1979, immediately after completing his M.Pharm, and successively worked as professor, dean, principal in University College of Pharmaceutical Sciences, Kakatiya University till 2007. Sir has an illustrious academic career with more than 30 years of research and teaching experience and 70 years of industrial experience. Sir mentored over 25 students for PhD and over 60 students for M.Pharm degrees. Sir published more than 200 research and review articles in reputed journals with high impact factor. Sir has more than 10 patents. Sir completed several projects sponsored by AICT, UGC, DST, etc. Later, he worked as a clinical pharmacology fellow in Hazelwood. He served at various capacities as director, senior director, president, vice president, and CEO in various pharmaceutical companies in the USA for the last 14 years. This showcases his professional expertise. He is contributing as his professional knowledge as a consultant to several pharmaceutical companies in USA. Yet present, he is a co-pioneer of 6S Pharma LLC, New Jersey State, and also serving as Chief Scientific Officer. Sir's professional experience exemplifies his academic and research expertise. Sir is a life member of several prestigious associations: American Society of Clinical Oncology, American Pain Society, American College of Clinical Pharmacology, American Association of Pharmaceutical Scientists. The list goes on, etc. Sir delivered several invited talks in international and national conferences. As a coordinate, Sir organized several conferences and also acted as chairperson in international and national conferences. Sir served as a member of advisory board and reviewer for Journal of Clinical Research and Regulatory Affairs and Asian Journal of Pharmacodynamics and Pharmacokinetics. Sir is a recipient of several prestigious awards. Sir received Fellow of American College of Clinical Pharmacology, FCP, Professor M.L. Corona Memorial Award in December 2016, Innovation Awards in 2014, Covidian Innovation. Sir, last, please, just. Sir is a founder and teacher. Actually, sir did not leave any field, and this implies that he is a co-founder and teacher of Sanatana Yoga Marg, a non-profit service organizations which gives instructions on yoga, pranayama, and meditation. With a great respect and salute for exemplary experiences and achievements, shall we welcome him with a standing ovation? I welcome you, sir, to deliver his keynote address on this auspicious day. Thank you. Thank you so much.
Good morning, everyone. Respected dignitaries on the day of the day, particularly Professor Roma, Professor Rajni, Dr. Narayan, who is a good friend of mine, and Joshna Rani, Vijayavati. In the audience, I see Professor Prasad, who has been a long-time friend of mine. I'm happy to see him. Actually, I didn't expect him to be here today, but really honored that he is here. He could make time. And Dr. Nagaraju, and many others, including Santrani. All these were my students a long time ago. I'm really fortunate to call myself to be their teacher. Because seeing the seeds that you sown growing into huge plants, or trees rather, including Dr. Surya Kumar, who was my first PhD student, is here today, and many others. Even if I don't mention the names, please don't think otherwise. And I am also very happy to have Miss Bindu Sharma from Bangalore, who is the CEO of Origin Solutions, who has been working with me on several groups whom I consult regarding drug development for converting them into patents and other useful documents. Since the time that I have for keynote address is fairly limited, I think it's about 4 or 5 minutes. So, but I reserve most of my comments related to the drug development and the translational research in the time allotted after some time. So please bear with me until then. But right now, since it is a keynote address, and I didn't expect Professor Uma to make such kindling remarks or comments. And that's why I always felt and feel, even today, it's imperative, it's very important, extremely important to have a social scientist in the midst. Okay, we need to have, because everything that we do has a lot of social implications. Instead of just individual or personal implications, we have a lot of social implications. And whatever we do, eventually, it has to reach the society. And so it's not just a science doing something, releasing a medicine and then expect profits out of it. That's not the intent of anything, particularly drug development. Therefore, it's nice to have some kind of good feedback from social scientists. And some of the points you made, in fact, they cover the actual topic today. We don't even have to continue the session further. Because she was talking about the translational research. I think she put everything in one sentence. That is bench to bet. So that is translational research. So whatever we do in the lab, whatever clinical work, preclinical work we do, that is in animals, in the lab, and then developing the drug molecule through a battery of techniques and technologies eventually reaches the patient and then the society at large. And that is what is called translational research. And what kind of things that you take into account and fill the gaps in between in order to make a drug really useful for the patient. So this kind of social consciousness is extremely important. And so with science without humanity or without humanities, is irrelevant. And that is why I think earlier, once upon a time, I'm not quite sure whether it was called science or not, but today universities do call social sciences. And apart from that, I want to make another remark. I miss Dr. Bharati today in the audience. In 2019, when I was here, despite her illness, she was here, and we had a good chat. And I really, really miss her. Wonderful lady, and a good pharmaceutical chemist. So I want to just take a few points, just 4 or 5 points, which I think should reach the ears of the administration. That's the reason why I would like to address these points here today. And then we are going to talk more about science, development, etc. in my actual lecture. The first thing is, she was also mentioning about artificial intelligence. Then she mentioned something about the collaborations and so on. So the first point is that today we are, we are flooded rather by huge amount of data. So we don't know what to do with it. So whether it is pharmaceutical research or any other research, particularly pharmaceutical research, we have been developing so much of thing, and there is a lot of enormous amount of data, and this data is intimidating. Okay, it's not just something that you could cope up with, and how far that data is relevant to any given purpose. So I think all of you understand, right, what data means. So tables, figures, in so much of things, and data without a purpose is useless. Okay, so you need to configure that data. You need to provide structure to that data. Understanding what data is called information. I think all of you know that, right? So what is the difference between data and information? So whatever data you have around, so much of it, some of it probably is useful for a given thing, and what the data is defined as information. And then what is the difference between information and knowledge? So if you ask what, then it is information. And if you ask how, it is knowledge. Okay, that means you are moving from one stage to the other stage. So this is popularly called as data, information, knowledge, wisdom concept. And it is also called a pyramid, the base of which is data. All of it is not relevant. Some of it is relevant, and that is information. And if you know what is the data, then it is information, and how that information could be utilized is the knowledge. So is that all? Is that enough? So artificial intelligence is something that could take to the level of information and to the level of knowledge, but it doesn't take to the top. And what is the top? Top is wisdom. That is, wisdom is something that provides insights. That means you need to introspect, you need to understand why a particular piece of knowledge, a particular piece of information could be used or utilized. That is wisdom. So most of the knowledge-driven technology is lacking wisdom. That means we haven't been utilizing our wisdom for developing technologies. That is why, we are in a haste of developing smartphones. That's great. Smartphones today, they are serving a lot of great purpose. But is it so? Is it 100% correct? Are we not suffering on account of smartphones today? And we blame everyone, including kids. Okay, I have a 3-year-old boy, and he always uses a smartphone. Who are you blaming? So this is the kind of problem we have. We have been knowledgeable. We have used the information technology so much, and have been able to develop, devices, tools, etc. But they are going to take away the human space. So that's the reason why we need to use our wisdom. Otherwise, eventually, we face problems. Of course, I could further elaborate on this, giving a wonderful story, but due to paucity of time, I would skip it for now. And one point I would like to take it to the authorities here. Both Vice Chancellor and Registrar are here. I'm not quite sure whether you have a legal cell on the campus. Even if it is there, probably deals with the issues related to the services and other problems. But I think an organization like yours should have a legal division or cell which can take care of the intellectual property. The universities can earn a ton of money. Several universities in the United States or in many of the developed countries in the West, they have been using these legal divisions where the councils advise, help, developing intellectual property. All of us, particularly those who are doing research at MPharm level or at PhD level or beyond as postdocs, you have been doing a good amount of work. I know as a faculty here, work for almost 30 years. I always felt that, we were always in a great hurry to come out with a booklet, call it a thesis, and have the viva was finished and degree awarded. But you are forgetting there is a ton of wealth of knowledge. Okay, sitting in those books, unfortunately, you cannot patent any of that. So you cannot harvest. Okay, that could be utilized in developing drugs that could help in mitigating several illnesses. So you work on medicinal chemistry project, or you work on a pharmacology project, you work on any kind of project, but unfortunately, it's not seeing the light of the day. You're not able to take it to commercialization just because of the fact that it's not being patented. So patenting is one of the most important things that has to happen, wherever research happens. So that is the reason why India is one of probably in the last seat. I think it is the 76th country or something like that. Probably Ms. Bindu Sharma would be able to tell that. So we are backbenchers as far as intellectual property is concerned. So I think this is one area where perhaps things could happen. And then interdisciplinary research. We are always sitting in watertight compartments. Pharmaceutical science is different from home science is different from computer science is different from some other science. So I think we are having an innumerable number of people here, scientists here, who have got fertile imaginations and minds, and they can be clubbed together. We can have deliberations, people attending seminars that are conducted by other divisions elsewhere. Likewise, we have got excellent technology incubators on this campus. This is one of very few campuses that has a technology incubator. I think there has to be good amount of interaction, and this kind of interdisciplinary kind of research helps a long way in taking to the projects to the next level. We learn a lot from other scientists. As a pharmaceutical technologist or a pharmacologist, what I know about the biology of a certain thing is just minimal. So that's where I could certainly exploit other scientists from other disciplines. And lastly, collaborations. So industry is starved, particularly India, and I'm going to talk this a little more, has been developing generic drugs, although it has the potential of coming out with new chemical entities. It is really shameful. Okay, in countries like United States, England, we have our scientists working there, starting from Dr. Ella Pragad Subbarao. I think many of you must have heard this name. He was the number one to bring out several drugs, including an excellent chemotherapeutic drug for cancer, that is methotrexate. And likewise, he contributed so much. But why we are not able to do anything here? Why we are not able to come out with even a single new chemical entity? So we got independence in 1947. So we don't have enough knowledge here. So this is mainly because of the fact that we are not having collaboration. We do not have proper kind of representation at the industry. And so there is an acute need for providing a platform where industry can interact with academia. Do you know that more than $200 billion of money is being exchanged between industry and academia in the United States alone? So this is the reason why so much of work is being done, particularly the seeding kind of work, the fundamental work, basic work, which eventually translates into applied research and bringing out molecules. The company where I worked last for about 17, 18 years, we collaborated or partnered with some universities. And then some of the technologies we brought directly from there. The professor of that particular science, and he eventually became an employee of our organization. So this would happen only when there is interaction between the two. When industry doesn't come to your doorstep, I think we should go and knock their doorstep. So that is possible. So be courageous and go and knock the doors of the industry and establish contacts so that we have more meaningful relationships, particularly for the young ones. So thank you so much. Probably I might have exceeded the time allotted, like 4 minutes, and I don't know how much time I took. Apologies once again, and thank you so much for giving me this opportunity of visiting your institution once again. Thank you.
Respected dignitaries on the dais, off the dais, and today's delegates, good morning each and every one of you for the conference. I take it as my privilege to present before you the introduction about Professor T.V. Narayan Garu, who is the stalwart and very active pharmacy professional in India. Really, I take it as my privilege to present before you about T.V. Narayana sir. T.V. Narayana sir is an iconic figure in pharmacy and very active pharmacy professional, as all of us know. And he has done his B.Pharm, M.Pharm, M.Phil, PhD from University Department of Pharmaceutical Sciences of Andhra University. And he has received the National Merit Scholarship and B.V. Patel Trust Memorial Scholarship for securing first rank in B Pharmacy from Andhra University. He has 35 years of teaching experience, research experience, and administrative experience. And currently, now he is the director of Vikas Institute of Pharmaceutical Sciences, Rajahmundry, Andhra Pradesh. And his active involvement in the spread of pharmacy profession in India and at global level is evident from so many of his active contributions in conducting various seminars, conferences at international level and national level, international honors that he has received, FIP Fellowship for 2024, CPA Fellowship for 2024, FFPA Fellowship for 2020, and he has organized Indian Pharmaceutical Congress twice during 2024 and 2016 in Visakhapatnam, and he has leadership roles, served as National President of IPA 2018 to till date, and currently now General Secretary for IPCA, and unanimously elected as President for SEER Pharma Forum, WHO, Southeast Asia region 2024 to 26, and he is the member of Board of Directors of Asian Associations of Schools of Pharmacy and former Central Council member of PCI and Ministry of Health, Family Welfare and Government of India. And he has contributed so much at global level. The notable ones are he is instrumental in bringing FIP World Congress in 2011 to India and IPSF World Congress in 2011. And one more thing, I just want to I just want to tell one thing is during 90s, I think whoever finishes M Pharmacy and PGs from the institutions, TV Narayana is the one person to remember to get a job. He is the one to provide the jobs for many of us from Andhra University and other people. Many of our friends knows, even myself in that. Thank you so much, sir. Now I invite TV Narayana, sir. Thank you.
Thank you, Dr. Joan Jyoti, for an elaborated introduction. Respected Honorable Chief Guest of today's international conference, Honorable Vice Chancellor, Professor Uma Madam, and Guest of Honor, Registrar of Padmavati Mahila University, Professor Rajini Madam, and my senior and good friend, Dr. D.R. Krishna, for his keynote address. And head of the department, my classmate, Dr. Jyotsna Rani, and organizing secretary Vijayavati, and the audience, my senior and good friend again, Dr. Prasad, Nagaraj, Dr. Varaprasad, and staff and students and researchers here. Good morning to everybody. Good morning. At the outset, it gives me immense pleasure to be here on the occasion of this international conference on recent trends in drug development and translational research organized by Institute of Pharmaceutical Technology, Sri Padmavati Mahila University. I think this was the second time I am here at the university in the 2005. Again, everybody is remembering Dr. Bharati, who is also my classmate. We were invited by her that time. We came here along with the Kokate sir and all. We came in 2005. And it is my second visit to the university. And I am live example for the development of the pharmacy profession taking place particularly in Maida University. Because I have seen from the raw when it was started in the shades, now we have developed the pharmacy department, and I thank the university authorities for giving importance to the pharmacy and developing the pharmacy, because I have seen the report what HOD has given, the way the pharmacy department is progressing. It is really my compliments and congratulations to all the staff who are working here and contributed for the development of pharmacy. And coming to the, because I represent Indian Pharmaceutical Association, I'll say a few words about IPA before touching this topic. Indian Pharmaceutical Association is the national professional organization of pharmacists engaged in various facets of professional pharmacy. It was established in the year 1939, before our independence, and the father of pharmacy, Professor M.L. Straff, is the founder who started the IPA. And I am the 27th president of it. We have headquarters at Mumbai. And IPA works for the benefit of students, teachers, and scholars. How we are benefiting the students is we organize National Pharmacy Week every year, World Pharmacist Day every year, because whatever you people are organizing, that is initiated through IPA. And we organize a national electrocution competition for the students to bring the hidden talents of the student and to give them suitable placement, because the NEC toppers, all the toppers were assured a job placement during the Indian Pharmaceutical Congress every year. And we are offering 40 lakhs worth of scholarships to the students throughout India through IPA, IRF scholarship. And we are not giving for the meritorious student, we are giving for the poor and needy student. And any universities, because we believe that the actual poor and needy students hails from universities only. Almost 40 universities were identified throughout the country, and every student will get 25,000 scholarship for all the 4 years of his B.Pharm. Even Mahila University can approach IPA for this scholarship. That's what, because when it was one of the university from the state of Andhra Pradesh, the students is getting all the 4 years the stipend. Almost 5-6 years. It is Dr. Vadlumudi Rao, who is my immediate senior and president, who introduced this scheme. That is one, we are benefiting the student. And for teachers, we are giving a travel grant. If any teacher paper has been accepted for a national or international to present their research or their research work, we are providing the travel grant. You can approach the IPA, that is one. And for researchers, the topic is here, the drug development and translational research. We have introduced IPA ACG award for innovation in solid dosage forms, that is one, and innovations in packaging. That is 2 awards we are giving, and with the cash prize of 1 lakh. These are some of the incentives we are providing from the platform of IPA, where you can get all the information from the IPA website. I encourage you people to make use of all the benefits, whatever we are providing through IPA platform. That is one. And coming to the topic today, drug discovery and translational research. Krishna sir rightly pointed out why we are not able to come out with one NCE. We may say we are number 3 in the world in pharma. We are exporting to 185 countries, and so many blah 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 we will say about our pharma industry development and growth. Other than vaccines, we are nowhere. We have produced only vaccines to the world. We have invented vaccines. That is the only development in our pharma. And particularly industry point of view, we have not come out with a single molecule till today. That was the fate of our Indian pharma industry. We have developed all our industry because of with our reverse engineering skills. Why means if to come out with one new chemical entity, it takes more than 15 years of research and $1.5 billion of expenses. But we don't know out of that 10,000 or 20,000 molecules screened, whether it may come into the market or not. And earlier, companies also not in a position to take risk. They have not invested on research. Now, at least with the government initiatives, almost all MNCs are investing 15% of their income on the research. So if in future, we may come out with new chemical entity. And frankly speaking, the actual research does not come from a research laboratory. It comes from university. That was proven all over the world. The actual research starts at university level. But the industry academia collaboration is the missing point here. That's where the universities are not able to contribute to the industry. Latest Covishield, even it has come from Oxford University, that vaccine. So why there was a lacuna here? That's why all the regulators, even Pharmacy Council of India is insisting for industry collaboration for every university or institutions here. The future it may come out. And I have seen a small country, Iran, when I attend the international conference. I will be sitting along with Iran. I and India and Iran, we are together there. Last 3 years, they are toppers in patents. More than 2,500, more than 5,000 patents. I asked them how it is possible. In a small country like not even 10 lakh population. They said the university, they will establish a research cell. The teachers will continue that research. Even the teacher retired, he will come and assist the next teacher, junior most, and train them to acquaint this research and continue the research. That's how they are doing. And even the students, they are taking the help. Even the student completes it, he can come and continue it, or he can delegate it to the next juniors. That's how they are continuing and getting the patents. This module, okay, it may not be possible here, but for the student point of view, but for the teacher point of view, it may be possible. That's what I want to suggest our pharmacists to go for the strengthen the research facilities provided by the university and be collaborate with the industry where we can come out with one blockbuster molecule. That's what I want to advise. And regarding the drug development, translational research, future is going to change because future drugs are not the tablets, capsules. Future drug is going to be a personalized medicine. Still, no need to waste our time on the routine formulations now because no one drug fits all. That was the slogan of the WHO. Because the Dolo 650 where we used during the COVID-19, it may not required. My body may not require 650, but we are uniformly taking. That's what the side effects of the drugs we are facing now. The future is going to be personalized medicine, genomics, biologics, biosimilars. So many things are going to change the role. As the role of pharmacist is going to be changed. That's why the research you can concentrate by using this technology of artificial intelligence. Krishna sir rightly pointed out. The artificial intelligence or machine learning, they will not give the formulation directly, it will give the data. When we are doing research, we used to spend almost 1 year for literature survey. That's time saving will be there with the usage of these new technologies. The new technology will not give the direct formulation. You have to adapt the technology for fastening of your research work. That's what I want to say. So concentrate on the latest development taking place, not on the conventional methods of research. implement all the what are the technology is available with us. In this, I greatly admire the great words told by Dr. APJ Abdul Kalam during the Chennai IPC. Technology is an important tool to give fast healthcare. Technology is an important tool to give fast healthcare. We have to use it. That were the great words told by him during IPC of Chennai. And about not only drug discovery, the practice point of view we are missing. That is, practice component is going to change the role of pharmacist in future. COVID-19, many of us witnessed how we suffered during the COVID-19, but blessing in disguise, COVID-19 proved the role of pharmacist a worth throughout the worldwide. If you take, many people will say, 8 lakh doctors worked during COVID-19. 10 lakhs health workers from government officials worked during COVID-19. Do you know how many actually worked? Who are the more number? It is pharmacists. More than 14 lakhs pharmacists worked during COVID-19. Throughout this pandemic period, when it was not recognized, we have sent a representation from IPA, and Prime Minister Modi ji and Amit Shah has sent a encouraging words to all the pharmacists. That's what we should feel proud to be a pharmacist. That's what COVID-19 has proved. Now, recently, government WHO has given a statement. After the COVID-19, don't think that our role is completed by coming out with Covaxin, Covishield and all. And WHO has pointed out 10 major global threats in future going to shake the world because of NCD, non-communicable diseases, because of coronary pulmonary disorder, because of alcohol, tobacco, air pollution. So many reasons they have given 10 global major threats are going to come. We should be ready with our preparedness to tackle these 10 global major threats. And at the same time, they have come out with 3 major projects that IP has signed all the 3 major projects. One is 1 billion people will be trained for universal health coverage throughout the globe. And 1 billion people will be trained, people means pharmacists, will be trained for health emergencies. And 1 billion people will be trained for the health and well-being. These are the 3 major projects. Even I want the young generation pharmacists to be involved in this project. They can write to our WHO headquarters in Delhi. Definitely you will be involved in such type of projects. University should be a platform to give training on these major projects. That's what I want to suggest on this platform. So finally, encouraging words for a pharmacist is, recently, last month, the WHO has given a statement about the role of pharmacist. Pharmacist, P stands for promoting health, P stands for providing health, P stands for protecting health, P stands for powering health, P stands for performing for health. So feel proud to be a pharmacist. That's what I want to tell all the generation who are present here. And beautiful words said by FIP President Dominic Jordan in the Glasgow conference is, 21st century is the century of pharmacists. 21st century is the century of pharmacists. And the pharmacist and the pharmaceutical scientist will be recognized as a competent healthcare professional by the society. With these few words, I thank the authorities for giving the opportunity to, I thank all of you for your patient hearing. I wish you all success in the future. Thank you.
Thank you very much, sir, for your valuable message. So your words demonstrates how deep knowledge you have in the subject also. So sir has mentioned that in future we are going to have the personalized drugs. So being a home scientist, always my concern is for the healthy diet. So always I feel that healthy diet minimize the risk of chronic diseases. But due to the change in the food habits and the lifestyles and the entry of so many fast foods in the market, we are facing lot of problems also, and we are depending upon the drugs. So we have to wait and see what the future is also. But coming to the today's topic about the recent trends in the drug development and translational research, as all our speakers have said, we stand at a pivotal, what is called moment in the history of healthcare. So recent trends in drug development and the translational research are reshaping the way how we approach the medicine. And these changes promise to revolutionize not only the treatment of disease, but also the very nature of how we perceive healthcare delivery. And all our speakers are also mentioned about the different tools that the artificial intelligence and machine learning are also revolutionizing the drug development pipeline. So by utilizing the what is called AI to predict disease outcomes, optimize clinical trial designs, and identify novel therapeutic targets, we can accelerate the development of new drugs while minimizing the risks and costs. So I feel that these tools, however, require substantial investment in infrastructure and the education, not only within the industry, but also in the public health system that will eventually implement these innovations. We have the tools to change the trajectory of medicine, but it will take collective action, vision, and commitment. So the future of the healthcare is now, and it's a time for all of us to step up and ensure that these innovations reach the patients who need them most. So I think the deliberations in this conference will definitely help all the participants to think in the right path and work in a successful manner. So I congratulate the Institute of Pharmaceutical Technology and the organizers and the other team members for taking up this international conference on this topic, which is very latent and which is very much essential for today. And I wish all the best for all the faculty members. Thank you. Thank you for giving me this opportunity.
My job is made easy by our convener. Yeah. My job is made easy by our convener and also president of this function. Already they expressed thanks in at every moment. And I take it a privilege on behalf of organizing committee to propose vote of thanks for the inaugural session of IPTCON-2024. First of all, we would like to thank our beloved Vice Chancellor, Professor V. Mumma Madam, Chief Guest of this IPTCON, for being with us and guiding in the right directions, who is the instrumental in the beginning of translational research and transdisciplinary research in the campus. And because of Madam only, we could come out of box of our pharmacy and we could reach different departments, medical colleges and other, and her guidance made us encouraged us towards translational and transdisciplinary research. Thank you, Madam, for being with us and in spite of her busy schedule of flighting to the abroad and her official assignment. Thank you, Madam. I thank our cool and dynamic registrar, Professor N. Rajni, Madam, President of this function, for being with us and for her continuous support and encouragement to conduct this conference. I take this privilege to thank our favorite teacher and today's keynote speaker, Dr. D.R. Krishna Garu, without whose we could not plan this conference, and for being with us and in his small schedule of India visit, he could made it possible and he could join us for this conference. Thank you very much, sir. I thank other stalwart of pharma profession, Dr. T.V. Narayana Garu, who is instrumental in the growth of pharmacy profession throughout the country and also global level. And he has joined us today for this conference. Thank you very much, sir. Though Sir, we have seen that sir throat is not good, but still you have made it possible, sir. Thank you very much, sir. Now I thank all the different institutions of pharmacy in and around Tirupati for joining us and registering as delegates for this program, and their management also for deputing them to this program. I thank our senior professors, Dr. KVSRG Prasad sir, Professor Nagaraj sir, Professor Santrani madam for their moral support and continuous encouragement throughout this conference and also throughout the department. Thank you very much, and madam and sir. I thank all the speakers for accepting our invitation and also joining and to enlighten on the topic to the delegates of this conference. And I thank the delegates from different colleges, especially from the SP Medical College, Dr. Roja Ramani, Dr. Lokesh from the SP Medical College. They have registered for this conference, and that made us this platform we can see now help us to have a good interactions and to develop our translational and transdisciplinary research, which is the theme of this conference. I thank all the students, faculty of IPT for their continuous support. I also thank our Dr. Surya Kumar Garu, who has started the startup ecosystem, and CEO of TBI for joining us on this occasion. I thank Central Network Facility of our university, who made it easy for the digital access, for making links and for providing video of this conference, which helps us to revisit the conference. I thank the Saveri Guest House personnel and staff of Saveri Guest House for arranging these facilities for this conference. I thank print and electronic media for their wide publicity. I thank one and all. Thank you very much. Thank you. On recent trends in drug development and translational research, we have learned a lot in the last day, yesterday, regarding this topic, and now we have our resource person. Excuse me. Yeah, some of them are joining. Just for 2 minutes, they are coming. Now we have Dr. Ritu with us from USA, who is going to talk on the entrepreneurship journey, and Jan 1 is first in class dual signal modulators for anti-inflammatory and endothelial stabilizing therapies. Now I request our chairperson, Professor Rajita is here, who is going to introduce Dr. Ritu. I welcome Rajita.
Yeah, good morning to all my friends and my dear delegates. Good morning, madam, dear Ritu madam, Dr. Ritu. Yeah, it's my privilege to introduce Dr. Ritu Lal, CEO and co-founder Gen 1E Life Sciences. Ritu Lal did master's degree in pharmacology from the University of Arizona, PhD in pharmaceutical sciences from the University of Maryland School of Pharmacy, and master's in management from Stanford University, MSX program. She has contributed more than 60 publications, book chapters, and presentations. She has 20 years of experience in biotech, pharma companies. Her passion is to bring healthcare products to patients in a cost-effective and efficient manner. In her role as a senior executive, she led preclinical and clinical development teams. She has filed more than 15 investigational, please give her a big applause, investigational new drug applications in various disease areas, including neurology, inflammation, virology, pain, and oncology. Two compounds under her leadership received new drug application from FDA and global approvals. Calatra for the treatment of HIV, Horizont for restless legs syndrome and post-herpetic neuralgia. Currently, she is CEO and co-founder of Gen 1E Life Sciences, a clinical stage in phase 2 clinical trials, biotech company accelerating novel next generation immunomodulatory therapies for rare and inflammatory diseases. I welcome you, madam. Thank you for joining with us. Thank you for accepting our invitation. Over to you, madam. Please, I have your talk. Just a minute. What's the talk? Yeah. Title, please, I request you to deliver your title, that is Entrepreneurship Journey Gen 1E's first in class dual signal modulators for anti-inflammatory and endothelial stabilizing therapies. Thank you so much, madam. Thank you. I welcome you to deliver. Over to you, madam.
Thank you so much for the kind introduction, and good morning to everybody here. I can see you guys on the screen. So welcome, and thank you, Dr. Vidyavati, as well as Dr. Krishna, for inviting me to present. And I will share my screen. Please let me know if you can see the slides.
Yes, madam. It is visible for us.
Okay, thank you. So as you guys mentioned, so I am Ritu Lal. I am the CEO and founder of this company called Genie. Genie, like the magical lamp. I don't know if you can see my pin here. It's like the magical lamp. And I will talk about my entrepreneurship journey today and our first-in-class dual signal modulators that we are developing for anti-inflammatory and for endothelial stabilizing therapies. And whatever I'm saying today is my opinion and my journey, so please don't take it as a word of the government or the FDA or anything like that. So I will talk about my background and my experience, how I went through the process working in the industry, getting approvals for the drugs that I worked on, Calitra and Horizont. Then I will tell you about what made me start the company, the mission and the birth of our company called Genie Life Sciences. And then a little bit about the learnings from we are based in Palo Alto, California, in Silicon Valley, and other experiences that hopefully I can inspire some of you to start being entrepreneurs as well. So, and by the way, this talk is pretty informal. You could wait till the end maybe to ask questions. That may be easier because this is a video conference. But definitely do ask questions. I would appreciate you asking questions. And no question is too silly or simple or anything. Just feel free to ask. So my background, I'll talk about all the way starting from, just like you guys, I have a pharmacy degree from University of Mumbai, and then came here to the US for my master's and then my PhD, working in the industry for many years, and then going back to school to Stanford, and then starting the company. So I'll tell you all about that journey and then the company itself. So I'm a pharmacist. I graduated from in those days it was called UDCT. Now it's called ICT Mumbai. You probably have colleagues there. And so finished that, then came here to get my master's degree from the University of Arizona and my PhD from the University of Maryland. I worked on what's called placebo effects. So when you take a medicine, especially a lot of the pain drugs, there is what we call placebo response. And I modeled the placebo response after taking naproxen. And surprisingly, what I found was 50% of your response is placebo. So it's not even related to the drug. It's just your internal body responding to the thought that you're taking a medicine. So that's really important to note when you're developing medicine, that there is a huge placebo response, especially when you're working on subjective clinical endpoints like pain or cognition and things like that. So that was the first part. And the second part of my dissertation was working on this drug called fampridine. That was being developed for multiple sclerosis. So I worked on animal models of MS, showing that the drug improved what's called visual responses, visual evoked responses. And yeah, glad to know that about 15 years after I worked on it, the drug was actually approved in the US for functional improvements in walking by the US FDA. So that was great. After finishing my PhD, and that is, by the way, the picture there is me right after I defended my thesis. You can see the rotary phone. We didn't have cell phones back then. That was the day I defended my dissertation a long time ago. After I finished my PhD, I worked, the first job was at Abbott Laboratories. It's in Chicago. It's now called AbbVie. And very fortunate when I started, I joined the company. The company was working on this drug called Kaletra for HIV. And this is the time during the AIDS pandemic in the 90s when people were dying. So I joined the company as a research scientist and was very fortunate to have excellent bosses who allowed me to participate in all of these discussions with the FDA and all of that. We were able to get the drug approved very, very quickly, and I'll tell you about that process. And now, of course, it saved 33 million lives and made a lot of money for Abbott. But in terms of what happened, I'll tell you a little bit more, given all of us are pharmacists, you will appreciate all of this a lot more. So lopinavir is the HIV protease inhibitor. So as I was saying, we had a lot of interactions with the US FDA, and we got the drug fast track, which is a designation that means the FDA will give you priority, as well as what's called priority review. So we were able to get the drug approved within 4 years, which typically takes 15 years. So that was important. And what happened with the drug was, initially when we started working on the pharmacokinetics of the drug, we found out very quickly it has a very high metabolism. It gets broken down by the intestine as well as the liver by cytochrome P450-3A4. And interestingly, by serendipity, we discovered a drug called ritonavir, which is also a protease inhibitor that Abbott made, giving very small doses of the drug, increased the blood levels of lopinavir more than 50-fold. So in fact, this is what we did. So I don't know if you can see this graph, but on the left-hand side, you can see the small circles. That is, if you give lopinavir on its own, it has very low blood levels, not even getting to the 50% inhibition that you require for antiviral activity. And then you give a very small dose, about 100 milligrams of ritonavir, you see the blood levels move to 50 times higher, just with the little dose of ritonavir. And in fact, that has now become the standard of care. If you know this drug called Paxlovid that was used during COVID, that also takes ritonavir, small dose of ritonavir. So any protease inhibitor that is given for antiviral therapy pretty much as ritonavir as their backbone, which is an intentional drug-drug interaction. So we were the ones who discovered that by accident, and now it's become standard of care. The drug was able to perform really well in people, and we were able to get approval, as I said, within 4 years, even before we had all the clinical data available. So in fact, at that time, we had only completed about 600 patients worth of data for 24 weeks, so 6 months, which typically would have needed 1 year and at least 1,500 to 2,000 people. We had not even completed the entire toxicology package, but we were able to get accelerated approval because of the huge unmet medical need. So I think importantly, one of the lessons learned for me was, definitely working on a serious disease with a huge unmet medical need, like during the AIDS pandemic. And I think something like that repeated during COVID has much better chances of getting FDA speedy approval and getting the attention. Getting the fast track as well as the priority review was super important. Very close interactions with the FDA, with very frequent discussions. We had a phone call with them every week almost, and what's called rolling reviews from the FDA on all the data. So even before preclinical study was completed, we had enough safety to go on to the next clinical study. All of that was important, and it was a great lesson for me. And I was very excited that the very first drug I worked on was successful and made history. And then, of course, it took 15 more years for the second and third approval. So that's when I learned it's not so easy to get a medicine to the market. So after Abbott, I moved on to Roche, another pharmaceutical company based here in California. And I worked in the inflammatory and antiviral division, worked on drugs for emphysema, HIV, hepatitis, rheumatoid arthritis, osteoporosis. This is actually my team at Roche. I don't know if you can see me, but I'm right here in the middle. This is a long time ago, almost 25 years ago. And I worked on these drugs called P38 kinase inhibitors. I worked on 5 of them, but unfortunately, not just us, but everybody in the industry was not successful in developing these drugs because of safety issues, or they would just stop working. So I just thought I'll take a moment and tell you a little bit about what these drugs were, what was the pathways, because this is going to be important when I tell you about our company, Genie. So P38 kinase, so kinases are chemicals, as you know, in our body, and they regulate many of the cellular functions. They are in every cell of our body. P38 alpha is a subfamily. There's 4 different isoforms, alpha, beta, gamma, delta. And when you have any kind of trauma, infection, or what have you, the levels of the P38 alpha are upregulated. They get we have phosphorylation, and that triggers the entire inflammatory process. In fact, at least 20 diseases of inflammation are related to increased levels of P38. What happened was we already knew that, and we knew it was an attractive target. The problem was the way we were inhibiting. So with the drugs that we were working on in the past 20 years ago were what we called catalytic non-specific P38 kinase inhibitors, and they blocked what's called the ATP binding pocket in a competitive binding fashion in such a way that they blocked the entire pathway. P38 alpha has inflammatory but also anti-inflammatory substrates, and by blocking everything non-specifically, there were off-target effects, and the drugs would just become toxic. We had secondary infections, we had liver toxicities, muscle toxicities, as well as in a couple weeks after the drug was administered, it would just stop working. 37 clinical trials across many different pharmaceutical companies, all of them failed because of safety issues or toxicity, as well as efficacy issues. So that was quite sad for me. I remember that experience, 7 years of my life, nothing to show. But it was a good lesson learned. It is difficult to get medicines to the market because of all these issues of safety as well as efficacy. After that, I worked on another company. I went on to a company called Xenoport with a very different business model. And this company was actually founded by a pharmacologist just like us. And it was a very unique way of developing medicine. So I worked on a drug called a prodrug of gabapentin. Gabapentin is neurontin that has been on the market for many years. And you probably know it because it is used for pain as well as for epilepsy. The problem with gabapentin is that it has very poor oral absorption. It's only absorbed in the upper GI tract, as well as has a very short half-life requiring 3 to 4 times dosing a day, as well as a very narrow therapeutic window. Because the drug is administered for people with epilepsy, the high peaks, because of having this sawtooth kind of shape in terms of the pharmacokinetic profile, the high Cmaxes would cause people to have safety issues, and the low troughs would cause efficacy issues. So that was no good, and it's been a problem. So what we did is we developed a prodrug, a very unique prodrug of gabapentin, which we called Horizon. And by doing so, this prodrug is now recognized by nutrient transporters all through the GI tract. And we converted the drug. So this is a figure of what happened with gabapentin. On the left-hand side, that the drug was only absorbed in the upper GI tract, causing the poor bioavailability as well as a lot of variability in absorption. By converting it into a prodrug called Horizon, our drug is now recognized by nutrient transporters in the entire GI tract, and therefore the drug is absorbed much better. There's no saturation. It has high oral bioavailability, and we were able to actually convert the drug from a 3 to 4 times a day drug to a once a day regimen, right here. With really dose proportional pharmacokinetics that you see here in orange versus the parent drug, which had a poor bioavailability as well as a dose saturated absorption seen here. So the bioavailability was increased almost twofold from 30% to about 75%, as well as the variability in absorption from a lot of variability from only 5% absorption with an average of 35%. Now we have an absorption of about 75%, almost 100%. So this drug, based on the pharmacokinetic properties and good efficacy, was easily approved by the FDA for both indications, restless leg syndrome as well as postherpetic neuralgia, neuropathic pain. And we used what's called a 505(b)(2) application process, which means that we relied on a lot of the historical information already available for Neurontin from the safety point of view, but because our drug had a different PK profile, we had to do a lot of the efficacy studies again. And it took only 7 years. Typically, it takes 15 years. We were able to get the drug approved for the first indication within 7 years. And the second approval came 6 months after that. So that was a good example of thoughtful drug development and how do you get drugs approved quicker and cheaper. But of course, in the US, unlike India, I think it's less of a problem in India. You have to make sure the insurance companies still pay for your drug, and that does become a problem sometimes with repurposed prodrugs. So that's something that I learned the hard way. After all of that, this is about 17 years post my PhD. I felt like at that point I had learned a lot about the drug development process, a regulatory process, FDA approvals, and all of that. But I felt like I needed to learn about the commercial side of things. So I applied to Stanford Business School here in Palo Alto. I was very fortunate to be admitted to the Masters in Management program. And I call myself a pharmapreneur, which may be a new term. It's basically a pharmaceutical scientist plus entrepreneur. That's how I call myself. And it was great to actually go back to business school because I learned, of course, I knew about the biotech industry, but to learn about the healthcare industry holistically in terms of all the other, the hospital side, the digital health side, health economics, all of the other things about the healthcare system, I learned about that. I also think I learned to be what I call being comfortable, being uncomfortable, which I think some of you may want to think about. So don't take up a traditional job. Think of doing things in a way that actually makes you uncomfortable, because taking a risk is actually a good thing, and you will learn a lot and you will probably enjoy the process as well. And that was great. I finished my master's in management. I also had a lot of fun in business school. This is us doing a flash mob way back. This is during the Gangnam Style days. If you go to YouTube, you can find this video. It has about 1 million views. So yeah, we had fun, learned a lot, but also had a lot of fun. And then after graduating from business school, This is when the birth of Genie happened. And I'll tell you what the problem statement was, why I decided to start the company and what we are trying to do here. So as you guys know, it takes at least $1 billion to get a medicine to the market, just one drug to the market. And it takes at least 15 years from the discovery to FDA approval. And that's if you're lucky. It's only a one, actually, it's even worse. It's a 1 in 100 chance to get a drug from discovery to approval. So why is that? Why is it we call ourselves biotech, but why does it take so long and why is it so expensive and why are the odds so against us? So my goal with starting the company was to use technology, use precision therapies to improve the chances of success, to make drugs cheaper, 10x cheaper, get the drug approved at least twice faster, and improve the odds of success. And by looking at the diseases that we tackle, both from the commercial as well as developmental attractiveness. So what I mean by that is, working on those diseases, for example, that of course is a huge unmet medical need. That's number one. And there is nothing out there that really makes it important, attractive. But then the models, whether it's preclinical, in vitro, whatever, are predictable of the human condition. So that's super important. If you work on, for example, for Alzheimer's, almost all the drugs work in the mice, but they don't work in people. So why is that? Same thing with oncology. So many of them work in the xenograft model, but they don't translate to the clinical. So how do you translate from preclinical to clinical? What are the biomarkers that you can use to make sure that you have translatability? What are the clinical and regulatory feasibility? Are they clear? Remember, I talked about the placebo effect. Are they clear, objective measurements that you can use for the clinical endpoint? Does the FDA or the regulatory body have a guidance? Do they help you along the process? Is there a biomarker you can use? And then the readiness, does your drug have the properties to, whether it's a formulation, oral delivery, or IV, does it actually get into the tissue? What is the metabolism? All of that is important. And then from the commercial attractiveness, of course, having the need, what is the competition? You don't want to develop yet another drug for blood pressure. It has to be something meaningful. And then do you have, I think it's really important, can health economics wise, is it something that will get reimbursed? Also then, more importantly, are there any FDA incentives? Can you do a quick proof of concept study? How do you use AI as well as precision technologies to get your answer quicker and know if your drug is working? So for all of those reasons, I started Genie to solve the drug development and approval problem. And we searched for the right technology. I spent about 1.5 years searching for the right technology. And lo and behold, I found it back to P38, the target that I worked on many years ago when I was at Roche. But these drugs that we are working on have a very different way of working, the way they bind to the target as well as how they work. They're making them super selective and specific. And the company launched out of 2 accelerators. One is called Y Combinator, the other is a Stanford accelerator in 2019. So that was the company Genie, and I'll tell you a little bit about the company now. I'll pause for a second if there's any questions before I tell you about the company. Should I keep going? All right, I will keep going because I don't hear anybody asking. All right. So Genie is the name of our company. We are a clinical stage phase 2 company accelerating first-in-class precision therapies for inflammatory and rare diseases with unprecedented efficiency. So we have taken our lead drug from discovery to a phase 2 clinical study in about 2.5 years, and with just seed spending, which typically, of course, would take at least 10 years, at least 100 million. So that's something that we do pride ourselves, and we want to do more of that as we go forward, and I'll tell you about how we do that. So I mentioned my background. We also have a small team. It's about 6 people, 7 people total, and we want to stay lean and mean. The technology was licensed from the University of Maryland, my alma mater. I'm also now adjunct faculty at the University of Maryland. The Professor Hasday and Professor Shapiro are the inventors of the technology. So Janya Bumkar is my co-founder. He happens to be my husband. He's on the tech side. He has already had 4 successful companies. This is his 5th. Dr. Mridul Mehta, who is our AI ML platform head, he's the one who has developed the platform. Dr. Maya Das is our clinician. Dr. Nate is our CMC person. Plus, we have other people on the team. Everybody on the team is very mission aligned. We all are very passionate about getting these medicines to the patient faster and cheaper. And we work around the clock. It doesn't matter whether it's Christmas, New Year, we work through every single day. So before I start about what our drugs do, I also think it's important to talk about what we are not. So we are not a typical AI company. There's many of them. They spend a lot of money and they have long timelines, but they don't have a lot to show in terms of how many drugs are really in the clinic, how many are in phase 2 or phase 3. We're also not limited to AI companies for drug discovery, which is okay, but then if you don't get the drug to the market, what good is it? And also importantly, we are not like the old generation P38 kinase inhibitors or substrate of that MK2. So we are very different in terms of our mechanism of action. So we are for unique biology. Our story is that our biology is very different. We are dual signal modulators, and I will tell you about the mechanism in a minute. We have a validated mechanism of action, which was recently published in the Journal of Pharmacology and Experimental Therapeutics. We talked about our unprecedented efficiency, and we hope to continue doing that, accelerating drug development using our AI platform. And I think it's not just the platform, it's, as I say, the 3 T's. It's the team, it's the technology, and the choice of target, which gets us to the 4th T, which is the time to the patient. And of course, because it's important, it's never one size fits all. It's all about using precision approaches for diseases. We do that, especially with those diseases which have heterogeneity. How do we get those medicines to the patient that we think will be the most responsive to our drugs? So our first drug, what we call gene 1124, has, as I said, completed a phase 1 study from early discovery to phase 2 in 2.5 years with just seed funding. We are in the US currently, and I hope someday to be in India. We're already collaborating with the head pulmonologists in India as well. Currently, we are in 5 of these clinical sites, including Mount Sinai, as well as WashU and Cleveland Clinic and so forth. We have received fast track designation, which is important. Remember, we talked about that for Kaletra. We also have fast track designation. That means the FDA gives us priority. We are also funded by the government with what's called BARDA. This is a part of the government that also funded the vaccines and therapeutics during COVID. So they're funding our precision therapy approaches, and I'll tell you about that. We're also in partnership with Vituity, which is a physician-led group. It's about 4,000 emergency room physicians, and they have an investment arm. They've invested in us, but they also help us as a voice of the customer. So we talk to them about what diseases, where are the huge unmet medical needs? Is this a product that would be useful to you guys in the hospital setting and so forth? We have completed our phase 1 study in healthy subjects in 48 people, knock on wood, with good safety, PK and PD. And while we're developing our drug for the first indication, which is for acute respiratory distress syndrome, ARDS for short, we also have a second indication that we've already talked to the FDA about already. This is our formulation. Actually, the drug substance is made in India, in Bangalore. And this drug product is very stable. It's a small molecule. The drug is in solution because the people who take it in the ICU setting cannot swallow. So it's an IV parenteral formulation. And currently we have 3-year room temperature stability of the product. So while we're developing our first drug, knock on wood, our second drug, which is called 1123, is also rapidly progressing towards the clinic. This is what it looks like. If you see the bottom, these are small tablets. This is the size of a dime. So I guess in India, I don't know if you still have 25 paisa, but that would be the size. And the second, you see the capsule also very small. So we have high drug loading, very good properties formulation-wise. And we're developing it for chronic oral diseases like muscle degeneration and so forth. And then we have a third drug, which is using another target called ERK. And we're developing those for oncology indications as well. So we have a strong portfolio of more than 28 patents granted. We have 21 plus novel compounds in our portfolio as well. So let's talk about what problem are we solving and then how are we going about, how does our drug work? So as I talked about earlier, kinases in our body, there's at least, they regulate 30% of the functions of our body. P38, MK2, ERK are some of those kinases which are dysregulated and cause pathogenesis for several devastating inflammatory diseases, and therefore have been attractive targets in the past. The previous generation drugs, as we discussed, were immunosuppressive. They had off-target effects, toxicities, and lacked specificity. And therefore, if we could figure out a way to bring back the homeostasis and its cellular function without suppressing the system, without suppressing the immune system, then we would be able to treat all these diseases, which is super exciting. So all of this began then, as we talked about, the previous generation drugs were binding to this ATP binding pocket shown here in the red. Our drugs bind to a very different part of the P38 alpha enzyme shown here. And the blue binding site shown here is where our drugs bind. So they bind in a different place. And this binding pocket is very far away from some of the anti-inflammatory substrates. That is important. And in fact, you can see one of our drugs binding inside the binding pocket right here. And the way our drugs bind is quite different. It's what's called non-catalytic, non-ATP, what's called type 4 interaction. So whereas the first generation, the old generation drugs bound to the ATP binding pocket in a competitive manner, shutting down the entire pathway, making the drugs immunosuppressive and therefore toxic, our drugs bind to a different binding pocket in what's called a type 4 allosteric interaction. They're substrate specific, they're selective and non-catalytic. So this was recently published. If you do a search on the Journal of Pharmacology and Experimental Therapeutics with the name of our company, you will find it. So I'll explain how our drugs work. So in the resting state, starting from the left-hand side, P38 alpha is in every cell of our body. 90% of the time, it's in the cytoplasm. MK2, which is a substrate, an inflammatory substrate, is inside the nucleus. When you have some kind of stress response, like an infection or what have you, P38 alpha gets phosphorylated or activated. And then what happens is this activated P38 alpha gets inside the nucleus and further activates MK2 and bonds into a complex. This complex then migrates out of the nucleus and out into the cytoplasm, and there it further activates or phosphorylates other substrates like HSP27, RBP, and then downstream of that, TNF alpha, IL-6, IL-1 beta. So these are all the inflammatory substrates that cause the disease. What our drug does is quite unique. What our drug does is it breaks that bond between the activated P38 alpha and MK2. leaving activated p38 alpha inside the nucleus. Remember, it was outside the nucleus when it was inactivated. Now it's activated and inside the nucleus. And by this process, there are some anti-inflammatory substrates inside the nucleus. Those get activated by our drug. And this is a good thing. So these are what we call MSK1 to CREB, and then downstream of that is IL-10. So this is a good anti-inflammatory pathway that we have now activated. Now the activated MK2 but uncoupled, it's not coupled with p38 alpha, gets out of the nucleus, but because it's not coupled with p38 alpha, it breaks down very quickly. And therefore, it's not very strong in activating HSP27 RBP and therefore TNF alpha, IL-6, IL-1 beta. So therefore, it dampens the pro-inflammatory signaling. So the mechanism of action then is upregulating the anti-inflammatory and then dampening the pro-inflammatory pathway. So something simplified like this. Our drug downregulates, doesn't inhibit, that downregulates, modulates, I should say, TNF alpha, IL-6, IL-1 beta, among others. It also stabilizes the endothelium, and then it upregulates MSK, and then downstream of that is IL-10, IL-1RA, USP1. So that is quite unique, and because we don't inhibit, but we modulate these pathways, we bring back the normal homeostasis. That is super important, and therefore we don't cause the same toxicities or what's called tachyphylaxis like the previous generation drugs did. So in summary, in stark contrast to the previous drugs, we have what we call dual signal modulators that avoid tachyphylaxis and avoid toxicities as well. On top of that, we do have an AI platform that we use to develop diseases and drugs. So we have about 21 plus drugs in our pipeline because of the unique mechanism of action of our drugs. So because remember I told you p38 alpha is involved in many different body functions based on how the drug interacts with the target. The drug could work on many different diseases. So for example, if it modulates MK2, HSP27, that pathway is important for ARDS, asthma, COPD, and so forth. Some inflammatory processes, if it decreases the cytokine signaling, like rheumatoid arthritis, IBD, Crohn's disease, and so forth. Also, these drugs could be important in muscle degeneration if it goes through these pathways, neurodegeneration, cancer, aging as well. So we use our AI platform to match disease as well as drug signatures, among other things, so that we run less number of experiments. Instead of running 50, 60 experiments, we just run 1 or 2 experiments. So that's how we decrease the time to development and get these medicines to the patient quickly and cheaply. So our AI platform goes from drug discovery to compound selection to clinical studies and back. So we have this end-to-end process where we use our data as well as data from different databases that have been highly curated to improve the chances of success of our programs. In addition, in the clinical study, so we also look at what we call AI endotyping, and I'll show you examples of that. And by doing so, we also improve the chances of success based on how the drugs respond to us in the diseases. So this is an example for ARDS, which is a heterogeneous disease. I don't know if you know about ARDS, but ARDS is a disease that, maybe I have a slide on that. Yes. So ARDS is a disease that was the lead cause of death for COVID, actually. So anytime you have influenza or bacterial infections where the infection gets so bad that your lungs are affected, you have pulmonary edema, that's when you have acute respiratory distress syndrome. Currently, there's no approved therapies for it, as well as there's a huge unmet medical need. There's 40% mortality rate with at least 3x more days in the ICU, and the cost per patient is at least $100,000 US. So in this disease, currently, as I said, there's no approved therapies. What we have done using our AI models, we were able to figure out a way to, just with clinical variables, these are 8 clinical variables, so we don't even need biomarkers that take a long time to analyze. We can figure out how badly or what is the seriousness of the disease, as well as the subject's mortality, by just measuring those 8 clinical variables. And we're doing so currently in our ARDS study. We can also check then if our drug, we think our drug will work for everybody, but we can actually check for phenotypes based on these clinical variables in our study. So BARDA is actually funding this work right now, and we hope this precision approaches will be useful in not just ARDS, but any other diseases in inflammation as well as immunology that we can use to improve the chances of success. So this is our portfolio. We have one, as I said, in the clinic in phase 2. We have a second drug in the same series that we are getting very close to the clinic now. We have 17 others that are what we call series 2. So they were informed by the data from series 1, and then we have 25 more behind that for P38 alpha MK2. And then for the second target, ERK, we have one that's in compound selected that has very good data in oncology and pancreatic cancer, as well as melanoma, glioblastoma, and then we have 18 more in the pipeline. So this is our pipeline in a nutshell. Our first drug, GEN1124, is currently in phase 2 in patients with BARDA partnership. Our second drug we're developing for chronic immunology diseases, as well as for muscle degenerative diseases, and we're close to the clinic, phase 1. And the third one is in oncology that we are getting about 1 year from the clinic now. So as I told you, ARDS is a terrible disease, costs just in the US, it's a $25 billion cost. In the US, it's about a 40% mortality rate. In India, I know it's even worse. During COVID, it was a lot worse, as you all know. So this is a problem worth solving. We really believe that our drug will help. We have a lot of good data in multiple animal models. So this is a model where we induced ARDS in mice using influenza virus. So we dosed mice with the influenza virus, and then when they were very sick, about 2 days later, we gave 5 doses of our drug every day, 1 dose a day, or we gave the vehicle. And what you see is, this is a survival curve on the left-hand side. None of the animals on the vehicle, not a single animal survived. They all died. So it's a lethal model. Whereas we saw greater than 5-fold improvement in survival. And also an improvement in what's called clinical score, where 0 is healthy, 5 is dead. So again, in the vehicle group in the red, nobody survived, they all died. Whereas in the drug group, in our 1124 group, yes, the animals did get sick, but after a few days, actually, even after the drug was stopped on day 6 here, the animals actually got better over time. So definitely the pharmacodynamic effect of the drug is longer than the pharmacokinetic effect. Importantly, we see lung function improvement. So we saw an improvement in oxygen saturation, where on the vehicle control, nobody survived. They didn't even reach the minimum threshold for survival. Whereas with our drug, we saw a great improvement in oxygen saturation on the last day of dosing. And if you follow the animals longer, they actually recover to almost 100% oxygen saturation. We also saw a decrease in the what's called the proteins, or in the bronchial alveolar lavage, the fluid inside the lungs, as well as a decrease in neutrophils. And in terms of the histopathology here, you see the picture here. On the extreme left-hand side, you see a normal lung with no infection. The second panel, you see an animal who had the infection, was given only vehicle. So you see the purple patchy lungs with fibrosis and necrosis happening. The third panel is with our drug. So this is On the last day of dosing, you already see a marked improvement. You see none of those patches. You see a lot better lung. And then if you follow the animals even after the last day of dosing, on day 14, which you see on the last panel, the lung looks normal. So it's almost the same as a normal lung. So this is pretty remarkable, and we were quite excited to see this data. This is also the published data. If you go to the Journal of Pharmacology and Experimental Therapeutic, this has been published. Quite exciting to see this data, and that was what prompted us to get to the clinic. We did complete our first in-human study in healthy subjects. So this was a double-blind placebo-controlled study where we dosed people with either drug or placebo, starting with a very low dose, again following the FDA ICH guidelines, with 1/10 the pharmacological dose, which is 50 milligram. And the highest doses we gave was 900 milligram twice a day. So that's almost a 40-fold dose increase for 6 days. So we did this study, completed it actually during the pandemic. And knock on wood, we have good data showing excellent safety, tolerability, pharmacokinetics, as well as pharmacodynamics, where we measured proteins. We measured 1,500 proteins in the plasma of these people after inducing with LPS, which is a lipopolysaccharide, an inflammatory agent. And we saw some really good pharmacodynamic data validating our mechanism of action, but also many other things, which I will share in a minute. So this is super important compared to the previous generation drugs, which had all these issues. Knock on wood, our drug did not have any secondary infection that required antibiotics. We didn't have any issues with liver injuries, so no ALT or AST increases, no muscle injuries, no severe rashes or dizziness. And really importantly, we have predictable pharmacokinetics and pharmacodynamics from mouse to rat to monkey to humans. In terms of the pharmacodynamic analysis, remember I told you we looked at 1,500 plus proteins in the plasma. And what we saw was quite nice. We saw a decrease in neutrophil activation, decrease in inflammation as we expected. Importantly, we spare or actually increase anti-inflammatory pathways like IL-10. So this is again validating our mechanism of action. We saw biomarkers that show that we stabilize the endothelium and the epithelium. We decrease apoptosis, decrease tauopathy. So this could be useful in neuroinflammation. We also have an effect in host-directed responses to viral bacterial infections. So that's why it would work for ARDS, pneumonia, sepsis, and so forth. So with all of this good data, we now are in phase 2 with our drug, knock on wood, and hopefully we'll have approval shortly after we complete. The second drug, as I said, is an oral indication. We already have shown what we call no tachyphylaxis. So this is an important point. The previous drugs, old drugs, stopped working, say, about 2 weeks into dosing. The reason for that was they would be inhibiting anti-inflammatory pathways, and that would cause the rebound or tachyphylaxis. Our drugs do not do that. We also have excellent oral bioavailability in monkeys, greater than 80%. We have shown some good animal efficacy in muscle degenerative diseases, and our goal is to be in the clinic next year. And as I said, the third drug with a different target but similar modulating effect for ERK is being developed for oncology. She hoped to be in the clinic after that, 1 year after that. So in a nutshell, in terms of how we started and where we are, we started out with a single organ, which is the lungs, acute indication in terms of ARDS and IV formulation. Now we're working towards multiple organs, chronic oral indications and rare diseases. And the goal is then, of course, to go towards even more immunology and inflammation targets in the next few years. So that is kind of our journey. I think it was good that we were able and so far have done a lot of good things. And I have to say, this is my first time as an entrepreneur. I got a lot of help from joining these incubators. One of them is called Y Combinator. The other is called StarTex, which is Stanford accelerator. So they were able to help in terms of how do you pitch, how do you raise money, how do you develop. So the development part, of course, I knew a lot of that, but then equally important is how do you raise money and how do you get to the next milestone and all of that good stuff. We have excellent partnership with the government, and they've been really excellent working with, as well as I mentioned, Vituity. So all of that helped us, and I hope this journey has been, discussion of our journey has been useful for you to hear. And I'll stop there with this quote from Phil Knight, who is the founder of Nike. This is a quote at the Stanford Business School. If you ever visit, definitely see this. So I'll read it to you. There comes a time in every life when the past recedes and the future opens. It's that moment when you turn to face the unknown. Some will turn back to what they already know. Some will walk straight ahead into uncertainty. I can't tell you which one is right, but I can tell you which one is more fun. Thank you. And feel free to reach out. This is my contact. If you take a picture of the QR code, you can also get to my LinkedIn profile. Thank you for your attention, and I would love to take any questions.
Thank you, Dr. Ritu. Am I audible?
Yes.
Thank you so much for sharing your passionate journey, especially at the point when in India people were really afraid to touch P38. If you ask me from my personal point of view, the dual mechanism, how you are targeting both the P38 and ERK pathways, it was very interesting for me because I'm a pharmacology person. So I was very much personally excited to listen to your talk. And I think in the audience, probably the pharmapreneur term has triggered most of the young minds who are sitting here. All are mostly UG students and the PG students in the first year and second year. So I hope the participants of this conference will have a strong impression of the word pharmapreneur. Because I've seen a live example how being a regular pharmacy graduate, you see the life journey of Dr. Ritilal, which was very, very impressive. And I should wish you all the best for your Genie 1124 success. We will be cheering from you from India that your 1124 will definitely make into market very soon, especially by fast track approach. Thank you so much, Dr. Ritu, once again. And we are expecting any questions from the participants. Do we have? Yes. I request Nitya to please pass on the mic. I'll do that. The participant will come here and ask a few questions.
Thank you.
Good morning, ma'am. My name is Shulekya. I'm studying M-Pharmacy second year in SPMVV. Firstly, I like to ask you two questions. First question is, what are the suggestions you give to a M-Pharm student to become a pharmapreneur? And the second question is, as a woman, how do you balance your personal life and a professional life?
Wow. Okay, I'll start with the first one, which is a lot easier. And it took me, of course, you heard my journey. It took me 20 years before I started the company. I think though things have changed a lot. So think about you're working on some project for your M-Pharm right now. You're working on a dissertation or thesis. Is that something that is useful? Is that something that you can patent, number one? Is that something you can start a company? That could be even start now. Why wait 20 years like I did? So think about those kind of things. So for me, the journey was longer because I learned a lot of things along the way. But I see that the younger generation is a lot smarter and they use their PhD or master's thesis to start their own company. So think about that if that's possible. Maybe go on to do your PhD. If there's any idea that's patentable, Make sure you get your good patent lawyer, or file a provisional right away. So that's one way. I did go to business school. I learned a lot in business school. If you have an opportunity like that, we were able actually to take ideas and form companies, learn, write a patent, pitch to investors. So I learned a lot of those skills also in business school. So if there's a way to get to business school after your MPharm, that's another way. The second question is to do, and also there are these accelerators. I mentioned Y Combinator and StartX, and Y Combinator for sure takes applications from India. In fact, they even interview in India. So definitely look into those kind of ycombinator.com. Those are really excellent accelerators that help you along the journey. In terms of balance, there is no balance. And as a woman, I can tell you that whoever talks about work-life balance, there is no balance. If somebody can figure it out, you tell me. So my life, my passion is work. My husband is my co-founder. So you can imagine we work around the clock because at the end of the day, the patients are waiting. For them, there is no Christmas, there is no New Year. So, but at the same time, having some kind of rest, mindful rest, exercising, eating healthy, all of that is important. But yeah, passion, when you're working towards a goal, you will be so passionate that it doesn't feel like work. It's enjoyable and it's fun.
Thank you, Mom.
You're welcome.
Do we have any more questions from the participants?
There's a question from Sridharala Venkata.
No. Dr. Raju has joined online and he's asking some question. Dr. Raju, please continue.
Namaste, ma'am.
Namaste.
Very happy to see your presentation. And in fact, I missed a couple of slides, but otherwise, whatever I've heard, it's very interesting and useful. One question is, if I understand correctly, the molecule what you have taken, in that class, the secondary infections and those are the challenges, and your molecule, you have addressed that. So the current studies, whatever you have run, are they sufficient to establish that the secondary infections are really addressed? Because in the real-time scenario, probably these are used for longer duration. So that correlation is already existed, or that needs to be monitored over a period of time.
Yeah, it's a really good question. Sorry, are you a student or are you a faculty? What's your background?
Yeah, I have a startup and I've registered myself for PhD, and I'm pursuing my PhD currently.
Okay. Congratulations on both.
Thank you.
So yes, of course, secondary infections and other liver toxicities, all of those are important because I've seen it firsthand already. And in the previous generation drugs, we could even see it in the preclinical studies, by the way, even in the monkey toxicology studies, we were able to see that. We didn't see it in our drug. I didn't mention that, but we were able to show that even in the toxicology studies, the highest doses we tested were the NOAEL with big safety margin. So that was one. The clinical studies, yes, they were short. They were only 6 days. For ARDS, that's all, you don't have to dose longer than that. In the chronic oral situation, for example, when we work on those diseases, of course, we will be testing that again, and that's part of the plan. But because the drugs work quite differently, I'm hopeful and optimistic that we will not see the same effects, which we, even for 1124, we did not see them already in the preclinical situation, and that was shown in clinic, but we will have to test it for the chronic indications as well. So it's a good question. Keep your fingers crossed. We will test it in the chronic situation with our second drug.
Okay. Now, the reason why I'm asking is I'm just trying to understand, are there any accelerated testings to understand
The toxicology studies are quite predictive for people, especially monkeys, and we've used those in the past. So that already tells you something. So sometimes the early signals come quite fast in preclinical studies. Other than that, I think you just have to watch what you're doing in the clinical studies. We closely monitor for things like that. Yeah.
Okay, thanks. Thanks a lot. And congratulations and all the best for your future trials.
Thank you. And you too. Congratulations and all the best for your startup.
Thank you.
At this point, when we are discussing about innovation and startup, I like to once again highlight that Mahila University has 2 incubation centers available, one specifically for women. We call it as Women Bionist Incubation Facility funded by Department of Biotechnology. Whereas we have one more technology business incubator funded by DST. So as Dr. Ritu was mentioning, younger generation is having it little bit easy to have their own startups with their PhD degree ready with them. And looking for
I think they're more innovative, they're smarter. I feel like the younger generation is so much more thinking about entrepreneurship and innovation. Yeah.
So we hope that trend also picks up in our own girls in this Mahila University.
Absolutely.
Probably I'll be seeing 1 or 2 pharmapreneurs who are sitting here. Any more questions we have for this session? Professor Rajita wants to talk. Please come on.
I think someone in online also has raised their hand. Uma.
Uma?
Yeah, you have that. Madam. Yeah.
Please go ahead.
Hi, Dr. Ritu. I just have it was a great presentation and it was quite interesting actually. So I do have a question. You mentioned like we are doing like clinical trials. So do you monitor those data like with any software based, or you just like, is it manual or like any software based kind
No,
of
we
SaaS?
use what we call Medrio for the data
Metrio?
management. It's actually a Stanford company that it's a data management software for the safety data. And then for the pharmacokinetic, pharmacodynamic, of course, we use the typical WinOnline. For the bioinformatics, there were 1,500 plus biomarkers that we looked at. We had our AI person work on it using their own software, R plus and all of that. So
Okay, all the statistical analysis also will be
statistical
done with
analysis.
a.
Yes.
Okay, got it.
But the data safety database is called Medrio. There's others as well, but that's the one we used.
But those are like FDA approved or just like those are FDA approved?
They're 21 CFR compliant.
Okay, got it.
So
Yeah.
it's
Thank you so
Friday
much.
night for me. I'm a little bit.
Yeah. I know, I can understand. Thank you.
You're welcome.
Madam, I wanted to ask a very general question. You have done masters in management from Stanford University. In what way it helped your career, and especially that I wanted to know. Yeah, you are the founder or co-founder for this?
I'm the founder.
Yeah.
Yeah.
Actually, in the biodata, it was mentioned co-founder. That's why I wanted to clarify
Well,
that.
it's me and my co-founder is Sojanya, so we're both founders. So I think we use the term loosely, whether it's founder or co-founder. But I started the company. So your question has to do with what Stanford. So I learned a lot. To be honest, for me, it was almost a life-changing experience to go back to school. First of all, 17 years post PhD, going back to school and learning was amazing for me personally. I think for me, it was interesting. I learned, of course, the finance, accounting, all of those things. Honestly, they were very easy. After the PhD, this was not difficult. But most important, I took a lot of classes in medical school and engineering school. So for me, it was also learning about the rest of the healthcare industry. So I took classes in digital health. I had a class called Biodesign Innovation, where we actually worked on an idea from an unmet medical need to iterating it into a device, to actually writing up the patent, writing up the business plan, pitching it to investors, and starting a company. So it was a very experiential way to do things, to actually start a company while you're in school. And that experience, that company I didn't take forward, but for me, that experience of how to do that whole process was very, very important. Also, the mentors and the professors that I'm still in touch with, I think all of that was helpful. So, and I think for me, it was also a little bit of, because I was older, when I went back to school, I was almost 40. The people I was taking the classes with were in their early 20s. And I would say something like, oh, some idea. And I would say, no, this cannot be done. And then the 20-year-old would ask me, why not? And then I would think, okay, actually, why not? So also, for opening up my mind, sometimes when you grow older, you think things in a particular linear way. And to be able to think in a non-linear way, to think of why not, was actually a good learning for me, to always think and listen to other people's ideas. You don't know everything, and the younger people might have other ideas that you don't. So that was good as well.
Oh, really inspirational, madam. Your talk is excellent, inspirational,
Thank
and
you.
enlightening to our delegates. Thank you so much.
Thank
And
you.
one more, I wanted to clarify, Gen 1E or Genie?
Genie. Genie, like the lamp, like, Aladdin and the magic lamp, Genie.
Actually, that why it is looking like one. It is not
Okay,
I.
so I will tell you the story about the name. So the N1 is a play on words for two reasons. N1 is because we work on precision medicine. So N1, it's it's that one patient is unique. Everyone is unique. So N1. So that was one reason. So Genie, it's GEN1E. That's a play on words. And the second thing, we work on a lot of times rare diseases, unmet medical need. A lot of times are these rare diseases. So that's where, again, small number, but big need. So that's how the N1, so GEN1E.
Okay, thank you so much.
You're welcome. Thank you for giving me the opportunity, and definitely reach out if you have any questions or you need any help. I'm available.
Yeah. Dr. Ritu, thank you very much for joining us and sparing your odd time, I can say, with us.
No
And
problem.
I also thank Dr. D.R. Krishna for introducing you for this wonderful occasion. As a formality, we have a certificate to you, though it may not be useful to you, but it will be
No,
memorable to you, I
very
think. You
humbling.
can
Very
remember
humbling. Thank you.
our institute as you have presented here. And if Dr. Saujanya is still in India, if
No, he
possible, I'll
actually came back. I will be in India in December, so maybe I can stop by, or I can give you an Indian address to mail it to. That would
It's okay,
work.
it's okay. You said he is going to visit Tirupati. I thought I can hand
We
over
are
to him,
hoping. We
even
are
though
hoping. He couldn't make it, unfortunately, but God's grace, I hope we can visit sometime soon.
it's okay, no problem. Everything will be all right. Don't worry.
Thank
And
you.
once again, thank you. And all our colleagues are here. And I thank
Thank
all the
you.
colleagues also for joining. Our senior professor, Dr. Nagaraju sir, is here,
Thank you.
and who is senior most in our department and sparing his time. Thank you, sir. And all the doctors
Thank
are
you.
also from medical college and other
Oh,
institutes of our university, different departments and different pharmacy colleges are here.
oh.
And all of them, I hope all of them have got enlightened with your, especially women pharma entrepreneur or pharmapreneur, as you said. Thank you. Thanks a lot
Thank you
once
again.
again, and we'll meet you and I'll
Okay.
contact you later. Thank you.
Thank you so
Thank you,
much.
one and all, and give
Thank you,
him a real
Doctor.
big round of applause. Thank you, and
Bye.
good night.
Thank you.
Yeah.
Thank you.
All right.
Bye.
No, now good night to them. Now we'll go to the next session. Please, Richa.
So next we have our online oral presentations. For this session, we have Dr. Sujata ma'am as chairperson and oral online presentation. Yes, ma'am. Yeah. Only we have 7 online oral presentations and online. And these are the list of the participants. Be ready with your presentations. First will be Kavyasri. Next, Uma Maheshwari, Meena Kumari, Venkatanukaraju, Shai Qaida, Muhammad Rafiq, and Shaima. Please be ready with your presentation in this sequence. And please note the instructions. You will have your presentation time for only 8 minutes and 2 minutes for discussion. And first will be Kavya presentation. Please start your presentation, Kavya. Share your presentation.
Sure. Thank you. Is my screen visible?
Yes, your screen is visible.
Thank you.
Please switch on the presentation mode.
Sure. Thank you. Good morning, everyone. Today, I want to give my presentation on development of a tumor-responsive nanoparticle-based multidimensional therapeutics against cancer. This work was done in Birla Institute of Technology and Science, BITS Pilani, Rajasthan. So coming to the introduction, according to the WHO Globocon data 2020, cancer is the most dreadful disease among all the diseases. Among different types of diseases, breast cancer was found to be the most malignant one. And though there were multiple treatment strategies available for breast cancers, such as chemotherapy, radiotherapy, and surgery, chemotherapy being the first-line treatment strategy for breast cancer, though it was found to improve only 10 to 3% of survival rate among the older women. The major reason behind the failure of these advanced chemotherapeutics is heterogeneous tumor microenvironment. Thus, the need of the R is targeted multidimensional therapy. Why targeted multidimensional therapy? Because the tumor microenvironment not only consists of tumor cells but also comprises of many other non-tumoral cells like cancer-associated fibroblasts, tumor endothelial cells, cancer stem cells, and tumor-associated macrophages. Among all these cells, macrophages are the most abundant immune cells present in the tumor, which are responsible for the immune suppressive microenvironment. These tumor-associated macrophages are majorly present in the M2 phenotype, which are helpful in the promotion of the angiogenesis as well as metastasis, and which helps in the suppression of T-cell responses. Thus, if we can activate these M2 macrophages to M1 macrophage, we can enhance the antigen cell presentation along with the anti-tumor T-cell presentation as well. So these activated macrophages can also have direct cancer killing capability as well. So how these tumor cells are taking the advantages of these macrophages? When tumor cells grow and proliferate, they secrete certain chemokines like CSF1 and CCL2, which helps in the monocyte to macrophage recruitment. These again secrete some cytokines like VEGF, which helps in the promotion of angiogenesis as well as metastasis. Further secretion of certain cytokines like IL-10 and TGF-beta helps in the reduction of the cytotoxic T-cell activation, which helps in the increased tumor burden. Thus, if we can activate these M2 macrophages to M1 macrophages, we can reduce this tumor burden completely. So what can be done for this? There are multiple options to target these tumor-associated macrophages. Among them, majorly used one are macrophage depletion, polarization to M1 phenotype, modulation of secreted factors, and immunogenic cell death. Among all these, macrophage polarization ones were the most abundant used one because these can be converted to the immune suppressive tumor microenvironment to the immune stimulatory one and enhance the phagocytosis as well as T-cell activation. So we have selected TLR agonist among all these drugs for our study. So if we can re-educate this tumor-associated macrophage from M2 to M1 macrophages, these M1 macrophages can secrete certain cytokines like TNF-alpha, ROS, and which helps in the reduction of the tumor burden directly. And these also can secrete certain cytokines like IL-12, which enhances the T-cell activation, which helps in the anti-tumor T-cell responses as well. Thus, we want to re-educate the macrophages from M2 to M1 phenotype. So our rationale of the study was to combine the chemotherapeutic as well as immunomodulatory agent. But most of the chemotherapeutic drugs which are present in the market are immunosuppressive in nature, so that are not helpful for our study. But there are some exceptional cases like paclitaxel and gemcitabine, which we have selected for our study. So the mechanism of action will involve when this anti-cancer as well as immunomodulator reaches the tumor microenvironment, this anti-cancer agent can produce the cancer antigens, and immunomodulator can activate the macrophages and dendritic cells, and these can further activate dendritic cells as well as both cytotoxic as well as T regulatory cells as well, which enhances the immune cell responses as well as kills the tumor cells directly. So based on this rationale, we have proposed two different objectives, that is multidimensional approach using chemoimmunotherapy using anti-cancer molecule paclitaxel as well as TLR agonist, that is resectimod, and the other one is pH-sensitive nanocarrier-based delivery of combinational chemoimmunomodulatory agent for cancer. So here we are proposing triggered release nanoformulation, that is pH-sensitive nanocarrier, because there are multiple barriers involved for the diffusion of the normal nanoparticles in the tumor, which are intratumoral pressure, extracellular matrix, and dense cell population, where when we develop pH-sensitive nanocarrier, these nanocarriers will be stable in the neutral pH, that is blood pH. Once they reach the tumor acidic pH, that is around 6.5 to 7.5, they can get released, triggered by the tumor acidic pH, and enhanced drug release can be observed because of the acidic tumor microenvironment. Thus, we want to prepare pH-sensitive nanocarrier over here. So initially, we have screened the drugs, that the TLR agonist as well as anti-cancer agent, and we have observed that increased TNF alpha, that is M1 macrophage expression with resectimod at 1 micromolar dose against macrophage cell line, and when we have treated it against tumor cell line in combination with paclitaxel, we have found that at 5 nanomolar dose with 1 micromolar dose of resectimod was found to be additive dose. And we have treated this against tumor as well as macrophage condition media, and we have observed improved cytotoxic cell death with the combination when compared to the free drug alone. We have further observed M1 immunostimulatory macrophage increased expression as well as reduced immune inhibitory marker expression with the combination treatment when compared to the individual treatments. So after conforming the M1 to M2 to M1 immune stimulatory macrophage marker expression, we have then thought of synthesizing the pH-sensitive polymer. Further, here we have chosen two different polymers, that is linear PLGA as well as star-shaped PLGA, and we have synthesized a pH-sensitive polymer by using polyhistidine, which has a pKa value similar to the pH of the tumor microenvironment, and we have synthesized the polymer by using the polyhistidine. We have further characterized by using FTIR and NMR, and we have found the successful synthesis of the polymer. After the polymer synthesis, we have prepared different nanoparticles, non-pH sensitive and pH sensitive nanoparticles. Total 4 different nanoparticles we have prepared, and we have found that star-shaped PLGA nanoparticles were found to have uniform particle size with enhanced encapsulation efficiency and increased loading content. And when coming to the pH sensitive size, we have observed that star-shaped polyhistidine nanoparticles were found to show increased particle size at acidic pH and almost similar size in the neutral pH, which says that pH sensitive size was observed and the drug is getting released at the acidic pH when compared to the neutral pH, which is also observed in the pH sensitive drug release pattern as well. Further, we have also observed the in vitro 2D and 3D culture based
Kavya, you have
studies.
last 2 minutes. Please wind up.
Sure. We have observed improved tumor cell death as well as macrophage cell death, and improved tumor spheroid penetration with the star PLGA polyhistidine nanoparticles when compared to the other nanoparticles. Further, we have observed improved M1 macrophage expression and reduced M2 macrophage expression with the star PLGA polyhistidine nanoparticles. Further, we have found that improved pharmacokinetic profile with the nanoparticles when compared to the free drugs, and the toxicity study was also found to be the drugs were non-toxic at the doses we have given, and the in vivo anti-tumor efficacy of the drugs were found to show reduced tumor volume and reduced tumor weight with the nanoparticles when given in the combination when compared to the individual drugs. And these were in vivo biodistribution study was also found to show with the star PLGA polyhistidine nanoparticle uniform distribution of the dye was observed at the tumor tissue when compared to the other tissues, and reduced tumor proliferation and increased apoptosis and reduced increased DCFTA, which is an indication of the ROS analysis, and reduced metastatic nodules was observed with the star PLGA polyhistidine nanoparticles. Overall, the star PLGA polyhistidine nanoparticles were found to show the potential and chemoimmunomodulatory effect against the breast cancer model over here. Thus, I want to thank, these are the publications we have published from this piece of work. Thus, I want to thank Professor Aniruddha Roy from Bichpilani, Rajasthan, Washington University School of Medicine, Padmavati Mahila Vishwavidyalayam for giving me this opportunity to talk over here now. Thank you.
Hello.
Yes. Am I audible?
Am I audible, Kavya?
Yes, you're audible.
I have only one question for you, Kavya. When you working were you performing
Kavya, your work was excellent, but I have one question for you.
Yes.
You are working with anti-cancer immunosuppressive models, right?
Yeah, it is not an immunosuppressive, yes, BALC model, yes.
Then why you have selected raw cells for in vitro studies? Is there any specific reason why you have used raw cells?
Macrophage cell, and because the immunomodulator, whatever I'm using, is a macrophage activator. It is a TLR agonist which acts directly on the macrophages.
Okay. Okay, Kavya, we can move to the next candidate.
Thank you.
Next, Uma, please present your PPT. Please switch on your video also.
Yeah, sure, Nitya ma'am. Is it visible, ma'am?
Yes, your poster is visible. Please switch on your video also.
Yeah, I did the video also. So, hi, this is Uma. I'm currently doing as a formulation scientist in Quality CDMO USA. So I'm here to present a poster presentation on my formulation work regarding the suppositories. Actually, I developed like some of the suppositories which are used for the rectal and as well as anal like disorders. So the basic drug I used was hydrocortisone acetate. So I developed did the formulation method development and also the dissolution studies for the suppositories for the hydrocortisone acetate suppositories. So actually what basically I did is the most difficult part was formulating the suppositories with the suitable vegetable oil was taken there were 20 to 30 trials I performed to make it a suitable drug release with the dissolution apparatus. So once I formulated the suppositories, later on I conducted doing the essays, content uniformity, dissolution studies, melting point studies, and those all. So the base it is under the trial. I did 10 trials of essay results, and so one of the suitable vegetable oil that is called as Stephen M. Wekobi M, which is a hydrogenated vegetable oil that was suitable for the hydrocortisone acetate to release the drug at the 360 point time. So I mentioned the results also for the dissolution. So I made a 1000 kg batch with a trial batch, and I did took beginning, middle, and as well as end samples and conducted the tests. So dissolution also I used both the paddle and as well as basketed, but the basketed one gave me the good results, averagely 90%, 104% at 360 minutes. But currently we are focusing to formulate more, adding silicon dioxide, BHT to enhance the dissolution within 130 minutes or 180 minutes. And also we are also focused on to develop in the dissolution media to change the media concentrations, and those are to give a good certain amount of release particularly at 60 minutes, 30 minutes, like that. So based upon the drug diseases, we are like currently focusing on to release studies on the dissolution, and also majorly the suppositories are like it avoids the first pass metabolism. So that is the reason we are like focused on to do the suppositories like right now in our company. And also we did essays, content uniformities. Content uniformity is basically a problem because the suppositories we formulate is like we need to fill into the shells only 2 grams. So the 2 grams should be uniformly mixed, and the hydrocortisone acetate should be present 100% in the 2 grams of the suppositories. So that is the reason the content uniformity played a major role, which was also a critical part. Only thing is like melting point and as well as the mixing helped us to achieve the content uniformity results, like for all the 10 suppositories which I collected in the beginning, middle and end. So all of the suppositories were given like 104 like 104 percentages of results. And the suppositories also like we are also developing like glycerin suppositories also for the future developments. And we used like our HPLC methods only for both all the assay, content uniformity, and the dissolution samples were collected. We performed all the purity testings on HPLC method, so with a suitable column. So based on this trial 3 at 360 minutes time point, I got a good results of 100%. And also we are right now it is time 0 results only. So we are also performing like accelerated and as well as long-term stability studies also to give the correct amount of release at the temperature effectiveness also, because it melts at the oil melts at 37 degrees, which is the rectal temperature. So that is the reason we wanted to perform the accelerated and as well as long-term stability studies also. These are some of the references I used for the suppositories. Yeah, thank you for the time. I couldn't hear you, ma'am. It's in mute.
And now I think I'm audible now.
Yes, yeah.
My first question for you will be why you want to develop suppositories of hydrocortisone acetate. Is there any specific rationale for that?
Actually, in our company, we had a request of for the pain relief. So we had a request to develop with the use with this drug itself. So that is the reason we choose this drug particularly.
So based upon the customer need, you are preparing this suppository.
Yeah, based on the customer need and the marketing products. Yeah.
Okay, Uma. Thank you. We'll move on to the next candidate.
Yeah, thank you, ma'am.
Next, Venkatanookaraju sir, please share your PPT and switch on your video and keep your presentation in presentation mode. The next, sir, is Venkatanookaraju sir ready with your presentation?
Yeah. Is it visible?
Yes, sir, it is visible. Please switch on your video and please stick on to the time. The time is 8 minutes.
Yeah.
Thank
you. Good morning, all. Thanks for giving this opportunity to present our research work. So the current work is related to development, optimization, and scale-up of innovative topical formulation of teriflunomide for the treatment of atopic dermatitis. So I'm doing this work under the guidance of Dr. Pavan Kumar. He is the assistant professor in Geetham College of Pharmacy, Hyderabad. So just to give the background, AD is a chronic inflammatory immune disease. Unfortunately, there is no cure. So exact mechanism of action is unknown. And available treatment options act by different routes or different mechanisms. Example, steroids, calcineurin inhibitors, JAK inhibitors, even biologics. They are all there, but they act on different modalities and none of them, they give the cure. That's why exploring different drugs to treat such chronic uncurable disease is the need of our for any industrialist or any academic researcher. So recently we have come across oral dehydrogenate inhibitors. They were explored for the treatment of atopic dermatitis. So oral components, these are reported to have significant systemic side effects, and they also have black box warnings. So because of that, what we thought is topical route being a safer route than the systemic route or the oral route. And also atopic dermatitis being a skin disease, a chronic skin disease, we thought, why don't we explore formulating teriflunomide as a topical formulation and see if it elicits its pharmacological action and whether it is safe or not. So in this journey, so far what we have completed is we have identified and optimized the key excipients used in the topical formulation. And apart from several parameters, we have studied the impact of Freesta on the target product profile. These being semi-solid formulations, Freesta is an accelerated testing to understand whether the formulation and the process are stable and they have any impact on the target product profile. Post that, we have scaled up the formulation and monitored the stability. And because we are seeing promising results, our next steps are we do the repeat dose preclinical studies as part of a regulatory requirement. And then we obtain the clinical study NOC to conduct the clinical study. So prevalence wise, it's many patients, many kids, especially they are affected by atopic dermatitis. Overall, across India or globally, if we see, more than 10%, 13% of the children are impacted, and about 7 to 10% adults are also impacted because of atopic dermatitis. So there is a huge market need for the products. So the selected compound is a teriflunomide. Teriflunomide is an active metabolite of leflunomide. Leflunomide is available as oral tablets for the treatment of rheumatoid arthritis, and teriflunomide also is available as oral tablets for the treatment of multiple sclerosis. But so far, there are no topical formulations in the marketplace for both of the components. So between these two components, we have selected teriflunomide because leflunomide also is going to immediately get converted to teriflunomide in the body, and active part or active moiety is teriflunomide only. Hence, between these two, we have opted for teriflunomide. So there are a couple of publications. They have given us the confidence that yes, these components, leflunomide or teriflunomide, they act on atopic dermatitis because these two studies, even though they are very small studies, like they have tested in 1 or 2 patients, but these studies have demonstrated that on long-term usage of leflunomide orally, they have seen significant improvement on the atopic dermatitis. So as long-term treatment is required and these components are having black box warnings like secondary infections as well as the hepatic disorders, we thought a topical formulation will help. So the first component in our formulation is a penetration enhancer, because any topical product when we design, it has to penetrate through the layers of the skin. So we have selected a penetration enhancer and studied the impact. And between these studied concentrations, we have observed that higher the concentration of the penetration enhancer, higher the release. So for further studies, we have taken 15% penetration enhancer in our formulation. And the second component is a thickening agent. So this we have studied in 3 different concentrations. And all these 3 different concentrations, they have resulted in physicochemical properties wise, more or less properties. Even under polarized light microscopy also, there is no significant difference. And the release profiles, IVRT, when we have studied for these 3 formulations, there is no significant difference in the flux or percentage drug released. So this confirms that in the selected range of the studied range of this particular thickening agent, there is no significant impact on the release profile. As I have mentioned, topical formulations, Freesta gives an accelerated indication about the stability. We have studied before and after. So even though there is a slight difference on the pH, but the consistency, grittiness, phase separation, and even the XRD, there is no significant difference. Even polarized light microscopy wise also, there is no significant impact before and after Freesta. And even the IVRT, the release profiles are overlapping. So Freesta is not having any significant impact even on the release profile. So basis which then we have scaled up this particular composition to a 5 kg scale. And even on the scale up, we have seen there is no significant difference. And from the scale up batch, we have taken portions of the particular drug and understand whether pH has
Excuse me,
any
sir, you have
role.
last 2 minutes.
Yeah, pH has any significant role. And our observation is at pH 5, the release profiles are much better compared to pH 6. So the same scaled up batch, 5 kilo batch size scaled up batch, we have studied for stability. We have data for 3 months so far, 3 months accelerated conditions wise. There is no significant impact on the SA or the pH values or the viscosities. And even the France diffusion data wise also, there is no significant difference. The flux initially is around 360 and 3 months accelerated is shown only around 320, which is not statistically significant. And the USP, the unique selling proposition of our product is this will be the first long half-life topical formulation for the treatment of atopic dermatitis. So we are yet to establish, but our hypothesis is either this formulation will be applied once daily or even once in alternate day, so that it gives a lot of comfort to the patient and improves the patient compliance. We have a granted Indian patent covering the concept. And in a very informal one patient study, we have seen that the patient has shown significant improvement in the atopic dermatitis within just 3 days of application of our product. Thank you.
I will be having one question for you, Mr. Raju.
Yeah, please, ma'am.
Teriflunomide, you have been repeatedly telling that it is having a black box warning.
Yes.
To avoid that, you want to develop it as a topical formulation.
Yes.
Do you think that by changing a formulation, we reduce the toxicity? Is it possible?
Yeah. So because if you have seen in one of the slide, pH has shown at pH 5, there is a reduced release compared to pH 6. So we have opted for pH 5, which is lower release, so that the systemic absorption will be lower.
So if the concentrations decrease, will that be sufficient for treatment?
Yeah, because the drug will penetrate through the layers of the skin. And when it is deposited in the skin, and this is a skin disorder, there is a possibility that it will reduce the or it will elicit the efficacy, and systemic side effects will be reduced.
For treating any condition, we should have a desired plasma concentration. My worry was, will this formulation will be able to reach that plasma concentration? If yes, will that be sufficient to avoid toxicity?
Yes, because recently we have completed phase 3 studies of one more molecule for similar kind of thing, that is psoriasis. And there also in that molecule, we have seen oral products are having these systemic side effects. And we have formulated as a topical gel. And we have completed phase 3 studies with promising efficacy, and there are no systemic side effects. So in the same lines, we are exploring this molecule also.
Okay, we'll move on to the next candidate.
Thank you, ma'am.
It's Meena Kumari. Are you there? Hello?
Hello. Am I audible, ma'am?
Yeah, you're audible. Start your presentation. You have 8 minutes.
Okay, ma'am. Good, respected madams. Good morning, everybody. Today I'm here to present my research work, a part of my research work. This is my title of presentation: Molecular Dynamic Simulation. Simulations of bioactive constituents from Lapha cylindrica. It is also known as sponge gourd with potential anticonvulsant activity. This is my abstract. Molecular dynamic stimulations have become a standard tool for investigating biomolecules based on docking scores. The aim of present study was to investigate selected active constituents, namely furanol, hexadecanoic acid 2,3-dihydroxypropyl ester, hexadecanoic acid methyl ester, and 4-H-pyran-4-one 3,5-dihydroxy 2-methyl, identified in the herb Lapha cylindrica. The goal was to evaluate the structural stability, protein dynamics, and the absence of major unfolding events through molecular dynamic stimulation studies. Next, introduction. Molecular dynamic simulations, a powerful tool for investigating biomolecular stability, dynamics, and interactions. Objective of my study is to analyze the structural stability, protein dynamics, and interactions of selected bioactive compounds from Lapha cylindrica using molecular dynamic simulation. These are the targeted proteins I have chosen for molecular dynamic study: carbonic anhydrase 2, PDB codes 2A1H, and 6OE0 were chosen as the targets for anticonvulsant activity. Next, materials and methods selected. The selected compounds from docking scores. These are the selected compounds: furanol, hexadecanoic acid, 2,3-dihydroxypropyl ester, hexadecanoic acid methyl ester, 4-H-pyran-4-one. Methods: docking studies, identified key bioactive compounds, identified key bioactive compounds with potential anticonvulsant activity. Molecular dynamic simulation parameters: time 100 nanoseconds, metrics RMSD, RMSF, SASA, radius of gyration, and hydrogen bonds. These are the results and discussions of this this work. Root mean square deviation. Here protein RMSD stabilizes between 0.1 to 0.25 nanometers, indicating structural stability. Ligand RMSD showed minor fluctuations in between nearly 0.01 to 0.07 nanometers, suggesting stable binding. Root mean square fluctuations, high flexibility observed in active and loop regions. Most regions stabilized with RMS values below 0.1 nanometer. Radius of gyration, initial fluctuations observed in between 2.2 to 2.25 nanometers. Stabilization below 2.2 nanometers over time, indicating a compact and stable protein structure. Next, solvent accessible surface area. Consistent SASA values indicated no major folding or unfolding events. Next, hydrogen bonding. Increased hydrogen bonds towards the end of the simulation, supporting the structural integrity. These are the graphs and visuals of furanol. Next, conclusion. Stability. Stability, furanol and hexadecanoic acid 2,3-dihydroxypropyl ester exhibited excellent structural stability, compactness, and compared to the standard anticonvulsant diazepam. Dynamics, protein dynamics suggest flexible regions are critical for biological activity. Further research is going on with promising compounds selected for further experimental and computational approach. This is my presentation. Thank you, ma'am.
Meena, I have two questions for you.
Thank you.
In one, you have taken a plant and you have selected few phytoconstituents from it. Am I right?
Yes, ma'am.
How you have selected only 4 because the plant is having number of phytoconstituents? What is the basis for selection of only those 4?
No, ma'am. Already I have chosen 18 compounds from that, madam. That is methanolic extract that contain 18 compounds, madam. For that 18 compounds, I have done docking result, madam. Docking performed, madam. From that highest docking scores, these compounds only given, madam. They have chosen for simulation studies, madam.
Okay. Then why you have selected carbonic anhydrase 2 as a target? Can you explain how it can be anti-epileptic? If a drug is acting on carbonic anhydrase, how it can be anti-epileptic?
So, madam, carbonic anhydrase 2, this catalyzes the reversible hydration of carbon dioxide, madam. It inhibit the inhibition of carbonic anhydrase in the brain leads to accumulation of carbon dioxide in the brain, madam. It causes inhibition of the discharge, paroxysmal discharge, excessive discharges are inhibited, madam. That's why it reduces
Kavya, wait a minute. You are mentioning carbon dioxide is accumulated in brain.
In brain, madam.
Okay, okay, Kavya. We can go to next candidate.
Sheikh Aida. Sheikh Aida, are you there? Hello? Yes, ma'am. Sheikh Aida? Mohammed Rafiq, are you there? Sheikh Aida, Mohammed Rafiq or Sheikh Shaima? Is anybody ready? Mute lo unna. Okay. So we have our all our online presentations completed, and I thank our today's session co-chairperson Dr. Asha Begum madam and chairperson Dr. Sujata madam. Now we have a tea break for 10 minutes, and we gather here for next session by Harish sir.
I request all the delegates to assemble in sharp 10 minutes. We'll start the next session within 10 minutes.